A very pleasant morning to all the delegates, participants, and viewers. I warmly welcome you to one day international webinar on emerging trends in pharmaceutical sciences, organized by the Department of Pharmacy, Chaitanya deemed to be University, Warangal, India, in association with Maibo Groups and Indian Pharmaceutical Association. On behalf of Chaitanya deemed to be University, I, Dr. Divya Ashmira, working as Assistant Professor, Department of Pharmacy, feels privileged to welcome you all to the inaugural session of today's international webinar. I would like to start the inaugural session by inviting distinguished dignitaries, guests of honor, and invited speakers. It's my great privilege to invite the chief patron of today's webinar, Dr. C.H.B. Purushottam Reddy Garu, Chancellor of Chaitanya DM University, Warangal, India. I heartily welcome to you, sir. I feel honored to invite the chief patron of today's webinar, Srimati Suvarna Devi Garu, Secretary and Correspondent of Vishwabharati Education Society, Warangal, India. Heartily welcome to you, ma'am. Now I take the opportunity with great privilege to invite the patron of today's webinar, Mr. E. Vikram Reddy Garu, Vice President of Vishwabharati Education Society, Warangal, India. A warm welcome to you, sir. Now I take the opportunity with great privilege to invite the patron of today's webinar, Dr. C.H. Satvika Garu, the Vice Chancellor, Vice President of Vishwabharati Education Society, Warangal, India. A warm welcome to you, ma'am. Now I feel honored to invite the chief guest of today's webinar, Professor Chidamodar Garu, Vice Chancellor, Chaitanya deemed to be University, Warangal, India. A warm welcome to you, sir. Now I take the privilege of inviting today's guest of honor, Dr. K. Veera Venkataya Garu, Officer on Special Duty, Chaitanya deemed to be University, Warangal, India. Heartily welcome to you, sir. Now I take the privilege of inviting the guest of honor, Professor M. Ravinder Garu, Registrar, Chaitanya deemed to be University, Warangal, India. A warm welcome to you, sir. Now I take the opportunity with privilege to invite the convener of today's webinar, Dr. B. Mallikarjun Garu, Principal and Dean Faculty of Pharmacy, Chaitanya deemed to be University, Warangal, India. A warm welcome to you, sir. Now I feel honored to invite today's invited speaker of session 1, Dr. A. Pramod Kumar Garu, pharmacovigilance expert and drug safety officer, ex-employer of Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India, Uttar Pradesh. A heartly welcome to you, sir. Now I feel honored to invite today's invited speaker of session 2, Dr. Satish Babu Natarajan Garu, Head Department of Pharmaceutics, Faculty of Pharmacy, Lincoln University College, Malaysia. A warm welcome to you, sir. Now I feel honored to invite today's invited speaker of session 3, Dr. Vasudeva Rao Avupati Garu, Senior Lecturer, Department of Pharmaceutical Chemistry, E-Learning League, School of Pharmacy, International Medical University, Malaysia. A warm welcome to you, sir. Now, I would like to request all to join the inauguration of the program by seeking the blessing of Almighty with a digital lamp lightning and a prayer song.
सरस्वती नमस्ते वरदे कामरूपिणी विद्यारम्भं करिष्यामि सिद्धिं भवतु मे सदा भवतु मे सदा
उतगमातिसेवितम् कपित्थजम्बुफलसारभक्षितम् उमासुतं शोकविनाशकारणम् नमामि विघ्नेश्वर पादपङ्कजम्
for joining the inauguration. Now, I would like to invite the convener of today's webinar, Dr. B. Mallikarjun Garu, to officiate the program with a welcome note. I welcome you, sir.
I am audible?
Yes, sir.
Okay. A pleasant morning to all. Respected Chancellor, sir, Chaitanya D.M.T.B. University, Dr. C.H.V. Purushottam Reddy Garu, Patrons Mr. E. Vikram Reddy Garu, Dr. C.H. Satvika Garu, Chief Guest Vice Chancellor, Professor G. Damodar Garu, Guest of Honor, OSD CDU Dr. K. Veera Venkataya Garu, Register CDU Professor M. Ravinder Garu, dignitaries, invitees, delegates, scholars, students, a extended warm and hearty welcome to one and all on behalf of Chaitanya D.M.T.B. University, Department of Pharmacy, to a virtual international webinar in association with Maibo Group and Indian Pharmaceutical Association with a theme of emerging trends in pharmaceutical sciences. Being under ever increasing pressure to compete in a challenging economic and technological environment, pharmaceutical and biotech companies must continuously innovate in their research and development programs to stay ahead of the game. External innovations come in different forms and originate in different places, from university labs to privately held venture capital backup startups and contract research organizations. The speakers of today's program, Dr. A. Pramod Kumar Garu, Dr. Satish Babu Natarajan Garu, and Dr. Vasudeva Rao Avupati Garu, has doctorate experience both in academic and industry, and hope they will enlighten us on this virtual platform. Once again, thank you all. Over to Divya.
Hello, Divya. Please unmute your mic. Dr. Divya, please unmute your mic.
Yes, sir. Thank you, sir, for your warm welcoming note. Now, I would like to invite the guest of honor of today's webinar, Professor M. Ravinder Sir Garu, to address the meeting. I welcome you, sir.
Thank you, Divya. Good morning, respected dignitaries, guests, speakers, below lecturers, research scholars, and my friends. Warm welcome to everyone. It is an honor and a great pleasure to be here to share my views with you. Pharmacy is a health profession that joins health sciences with pharmaceutical sciences and plans to ensure the protected, successful, and reasonable utilization of drugs. Pharmaceutical science combines a broad range of scientific disciplines that are critical to the discovery and development of new drugs and therapies and includes versatile areas like drug discovery and design, drug delivery, drug action, clinical sciences, drug analysis, cost effectiveness of medicines, and regulatory affairs. It can be a key driver to revenue and a critical component of managing the health of consumers. Over the years, pharmaceutical scientists have been taking part in discovering and developing innovative drugs that save lives and improve the quality of life by working as pharmacists, doctors, as researchers, and professors at universities, as regulatory scientists for agencies like the Food and Drug Administration, and as a research at the national laboratories such as National Institute of Health and in various pharmaceutical companies. The work of pharmaceutical scientists doesn't just help relatively healthy people maintain their well-being. It gives people with serious illness the chance to regain their health. India's pharmaceutical sector forms a major component of the country's foreign trade with attractive avenues and opportunities for investors. India supplies affordable and low cost generic drugs and millions of people across the globe and operates a significant number of United States Food and Drug Administration and World Health Organization good manufacturing practices complaint plants. India occupies an important position in the global pharmaceutical sector by providing generic drugs globally at the most affordable prices. Indian pharmaceutical sector supplies over 50% of global demand for various vaccines, 40% of generic demand in the US and 25% of all medicines in the UK. Globally, from having a 5% share of the market in India and global pharma at 95% in 1969. Indian pharma is at 85% share and global pharma at 15% in 2020 due to several market segments like generics, vaccines, biologics, and others. India has shown excellent work on international cooperation by sending vital medicines to the world during the COVID-19 pandemic. Pharmaceutical sector of government of India is emerging into self-reliant in critical active pharmaceutical ingredient as well as medical devices. Pharmaceuticals industry is not only a great source of innovation and employment generation, but it has also become a global healthcare provider in need. Thank you, and over to Divya. Thank you, Divya.
Thank you, sir, for your valuable words. Now, I would like to invite the guest of honor of today's webinar, Dr. K. Veeravenkataya Garu, to address the gathering. I welcome you, sir.
Thank you, Divya. Honorable Chancellor Dr. C.H.V. Purushottam Reddy Garu, esteemed co-chairpersons Sri E. Vikram Reddy Garu, invited guest speakers were the eminent scientists Sri Dr. A. Pramod Kumar Garu, Dr. A. Satish Babu Natarajan Garu, Dr. Vasudev Rao Garu, our beloved Vice Chancellor Professor G. Damodar Garu, my colleagues Professor Ravinder, Registrar, Professor Mallikarjun, Dean Pharmacy, Dr. G. Kumaraswamy, Organizing Secretary, and Dr. Divya Ajmera, Coordinator, and other committee members, and distinguished participants and ladies and gentlemen. At the outset, I congratulate the organizers for conducting this international webinar on emerging trends in pharmaceutical sciences. On the advice and encouragement given by our Chancellor, Dr. Purushottam Reddy Garu, our university has been organizing various webinars, virtual workshops, faculty development programs in various subjects regularly. I am given to understand that there is a overwhelming response from the participants It is learned that it's more than 2,000 participants have registered, not only from India but also around the globe. Now participation, the registration is in progress. I think the progressive participation is also over. Since you are all expertise in pharmacy, I need not emphasize the importance of pharmacy in human lives. The services of pharmacy scientists cannot be measured in any terms. I'll quote one example here. At the time when the coronavirus started engulfing human lives in the entire globe, only pharmacy scientists came to the rescue by inventing vaccines and saving the human lives. This is only one example I quote. Developing and bringing new medicines that improve the quality of the life of the patients around the world. And our registrar already gave you the importance of pharmacy in India. But as an economic, the student of economics, I give the how the pharmacy contributed to the world. India's economy, it has contributed around $37 billion during 2019 and 20, and it has contributed to GDP directly 1.5% and indirectly 3%. Such is the greatness of the pharmacy scientists. And this is a program of only 3 hours. I think the patients, the participants will definitely gain some knowledge through this webinar. The speakers are of great eminence in their subjects. Therefore, I expect and hope that this webinar will be a great successful. I wish the webinar a great success. Thank you, Vanda.
Thank you, sir, for your kind words and wishes. Now, I would like to invite the patron of today's webinar, Mr. E. Vikram Reddy Garu, to address the gathering. I welcome you, sir.
Thank you. Good morning, one and all. A warm welcome to everybody for the international webinar on emerging trends in pharmaceutical sciences. It's another feather in the hat from the pandemic time started. The number of webinars Chaitanya has been planning and has been executing it successfully has been very commendable. And the kind of speakers and the industry leaders that are coming and sharing their knowledge is truly, truly inspiring for the students, for the faculty, and the community. I would like to invite, welcome, and also thank Dr. A. Pramod Kumar Garu, Satish Babu Natarajan Garu, Dr. Vasudeva Rao Avupati Garu, and all the team, the committee, the organizing secretary, and the organizing committee for this today's event. Today we have already heard from the registrar who spoke about, who gave a brief on the situation and the current trends and how the pharmacy and how India is contributing to the rest of the world, not only to the country. And our OSD Veera Venkataya Garu has also precisely described about the GDP contribution and the industry performance on today's date, the way it is growing and the way we have contributed in terms of vaccinations or research, also in trending and inventing new kind of drugs or methods that are in need of the hour today. I would like all the students to have a patient ear and follow the entire program today and gather the information that is needed and shared by the industry experts and have a wonderful journey. Thank you, one and all.
Thank you, sir, for your valuable words and wishes. Now, I would like to invite the chief guest of today's webinar, Professor G. Damodar Garu, to address the gathering. I welcome you, sir. Okay, meanwhile, we'll have an introduction of our institution with an AV.
Chaitanya deemed to be University, Warangal, Telangana. PhD programs in engineering, pharmacy, sciences and business management. Courses offered: engineering, pharmacy, PharmD, MSc, MCA, MBA, BSc, AI robotics, data science, BCA, BBA, BBM, BCom, industry integrated courses, BSc, computer science, cognitive systems, BCom business process management. Applications are invited for entrance test and admissions. మరిన్ని వివరాలకు chaitanya.edu.in కి లాగిన్ అవ్వండి.
Okay, thank you. Now, I would like to invite the chief guest of today's webinar, Professor G. Damodar Garu, to address the gathering. I welcome you, sir. Okay. There is a connection problem to our chief guest. So meanwhile, we'll have another AV regarding our institution. Please bear with us. Thank you all.
Where the heart is fearless and the mind is expanding along with the universe.
Where the eyes are empowered by thee to see the world differently each day.
Where existence is relentlessly striving to find its purpose.
Where the learning is beyond the horizon of intelligence and the walls of age or educational space.
Where imagination is unlocked by using the keys of reason.
Where the train of thought has not lost its tracks into the shackles of darkness. Is where life begins.
We create an opportunity for a new beginning.
Come, start living with us. Chaitanya Group of Colleges. It's an establishment for casting dreams to build a thriving reality. Chaitanya celebrates 25 glorious years in shaping the future of our young minds.
Okay, thank you all for bearing with us. Now, I request Mrs. V. Srujana Garu, Faculty of Pharmacy, Chaitanya deemed to be University. Thank you, sir. Thank you, sir, for accepting our invitation.
Am I visible and audible?
Yes, sir, you are audible. Sir, I request you to address the gathering, sir. Welcome you, sir.
Am I visible and audible?
Yes, sir, you are audible. Please go ahead, sir.
Is my voice audible?
Yes, sir, it is audible, sir. It is audible.
Sir, your voice is audible, sir. You please go ahead, sir.
Shall I?
Yes, sir.
Okay. Thank you, Divya. Esteemed Chancellor, Sir, Dr. CHV Purushottam Reddy Garu, Madam Swarna Devi Garu, Dynamic Co-Presidents, CE Vikram Reddy Garu, and Dr. CHV CH Satvika Garu, OSD Dr. K. Veera Venkataya, Registrar Professor M. Ravinder, Organizing Secretary Dr. G. Kumaraswamy, Convener Dr. V. Mallikarjun, Coordinator Dr. Ajmeera Divya, 20 members of the organizing committee, and 2,000 participants of this webinar. Namaskar. I am glad to know that the Department of Pharmacy is conducting two important academic events this month in addition to their regular academic pursuits. Last week, 5-day National Virtual Faculty Development Program on a current topic titled COVID-19 and Global Health was conducted with 5 eminent speakers. And this international webinar on emerging trends in pharmaceutical sciences is now being organized in association with MIBO Group and Indian Pharmaceutical Association Warangal with very good response. It's as some of you know, we have so far conducted 13 webinars on topics like how to use Google Forms, Yoga for Health on International Yoga Day, then on human values, emerging trends in genetic engineering and biotechnology, globalization and its impact on service sectors, statistics, role of rubrics, testing a hypothesis in the fields of science and management for the enhancement of validity, emerging trends in chemistry, national education policy 2020, then vigilance awareness, Our 11th webinar was on higher education opportunities in Germany. 12th one was on research methodology in life sciences. And 13th one was on yoga and health. And this is the 14th webinar we are conducting today. I welcome three subject experts, Dr. A. Pramod Kumar from UP, Dr. Satish Babu Natarajan from Lincoln University, Malaysia, and Dr. A. Vasudev Rao, International Medical University, Malaysia, to this important webinar today. Our Chancellor, sir, encourages us to conduct some more webinars with national and international experts to benefit all of us on different themes. We are moving in that direction. So I congratulate the organizers of the Department of Pharmacy on conducting this webinar today. I also appreciate the technical support given by Dr. J.S.N. Koteshwar Rao, Director of MIBO. I wish all of you all the best. Thank you. Over to you, Divya.
Thank you, sir, for your kind words and best wishes. Now, I would like to request Mrs. V. Shrujana Garu, Faculty of Pharmacy, Chaitanya deemed to be University, Warangal, India, to introduce the speaker of session 1. Over to you, Shrujana.
Good morning, everyone. I welcome you all to one international webinar on emerging trends in pharmaceutical. Department of Pharmacy, Chaitanya deemed to be University, Anakonda. Good morning, everyone. I welcome you all to one-day international webinar on emerging trends in pharmaceutical sciences, organized by Department of Pharmacy, Chaitanya deemed to be University, Hanamkonda. It is my pleasure to invite today's session 1 speaker, Dr. A. Pramod Kumar Garu, on overview on pharmacovigilance in India. Dr. A. Pramod Kumar, M.Pharmacy, Pharmaceutical Practice, PhD. He working as drug safety scientist for CRO based at Mysuru as a literature specialist. He worked as pharmacovigilance expert for NCC PVPL, Indian Pharmacopoeia Commission, Ministry of Health and Family Welfare, Government of India, Uttar Pradesh. He also had 2 years of experience in academics. He had published 35 papers in peer-reviewed journals and had attended 60 national as well as international conferences. He has expertise in sensitization of HCPs on PV, establishing a PV setup in hospitals, signal detection, and literature search. So now I would like to invite Dr. A. Pramod Kumar Garu. Welcome you, sir.
Thank you, Sujna, for that nice introduction. So before I go to my presentation, I would like to thank all the organizing members. So to name with Purushottam Reddy sir, Chancellor, Chaitanya Deemed University, and Chief Guest of today's conference, G. Damodar sir, Vice Chancellor, Chaitanya Deemed University, and also the guest of honors, Veeravenkataya garu and Ravindra garu, for organizing such a good conference in association with the MIBO group of Koteshwar sir and his team, and also all the office bearers of Indian Pharmaceutical Association, Warangal. So thank you all for inviting me and to provide this opportunity to interact with you. So please let me know once you are able to see my screen.
Yeah, Dr. Pramod, it's visible.
Yeah, thank you for the confirmation, sir. Yeah. So my next 1 hour of presentation will cover an overview on the pharmacovigilance program in India and what is its history and how it is taken away forward. So this is my disclaimer. The views and opinions expressed here are purely based upon my experience, and I am not representing any particular association, and it is not affiliated to my previous organizations or my current organizations. And I also
Yeah.
have no conflicts of interest in presenting this data. All the
Sorry for
information is
interruption.
yeah.
Sorry for the interruption, Dr. Pramod. There is some noise from your end. Maybe will you please reattach that audio?
Sure. So is it clear now?
Still some disturbance I am facing. Will you please remove it and connect it again? Earlier it
Sure,
was very
sure.
clear in the morning. Is there any fan in your room?
No.
Some air noise or some connection noises. Will you please
Okay, let
remove
me
that
try with
jar?
another. Yeah.
That's fine. Or with open audio you can try once.
Is this good enough?
Okay, better than earlier, but there is some disturbance.
Okay.
Is there any work going on nearby?
No, sir. Nothing is clear.
Okay, it is like some utensils sound I could catch.
Okay.
Will you
So
please
to start.
rejoin? Will you please rejoin?
Sure.
Because too much noise is coming. Yeah, Dr. Pramod, will you please speak something?
Yeah, hope it is clear.
Yeah. Okay, will you please share your screen and we will start, and meanwhile I will let you know.
Sure.
There is some issue with the audio jack, I think so. Earlier morning it is very clear. When you remove that audio jack and
Yeah, I have replaced it with another one. So still you are having same.
no, it's laptop, no? Is it laptop
Yes.
or desktop?
Yeah, it's
Will you please try open audio?
okay. So is it clear now?
Somewhat okay, but there is some issue. Have you? Is there any other audio open? Like YouTube Live is open in another screen, another window?
No, nothing is open.
Okay, will you please share your screen and we will see.
Okay. So shall I go ahead?
Yeah, 1 minute. Still we are getting some noise. Sorry to say that.
Okay. So it's better I will connect with.
Remaining screens and all audience, they are fine. I don't know. It's like some utensil sound or something like that. No, now your voice is also down. Is it possible to try with any other device, either mobile or something? PPT you can drag from this laptop, and audio you can try from mobile. That is one option we can try. Okay, dear participants, unfortunately there is some technical issue from speaker side, but it is very clear earlier when we have tested it, but unfortunately it happened. Excuse for that. And we requested the speaker to come with a new device. Let us give us 1 or 2 minutes time, such that it will be rectified. Thank you. Yeah, Mr. Srikanth, we requested that part also, but the speaker is saying there is no disturbance from his side. Maybe some technical issue, maybe because of audio job or something. Let it be. Let us give 1 or 2 minutes time. He is going to rectify. During testing mode, it's very clear, but just now only it started the disturbance. Let's bear with us because it's obviously possible sometimes as a side of disturbances. Let's bear with us. Hello, Dr. Prema.
Yeah. So can you hear me now?
Yeah, it's clear. Yeah, your
Okay,
voice
do
is
let me know
clear.
once.
Yeah, please
Yeah,
share
thank
the
you.
screen and we will continue. Yeah, thank you.
Sure.
Sorry for the disturbance. There is some issue.
No
That's why.
problem.
Yeah.
Okay.
Now it's very clear. It's very clear.
Okay. So do let me know once you are able to see my screen. So I'll start with my presentation.
Sure. It's just, I think, not at. Fine. I got it. Yeah.
Okay. Thank you.
Yeah, at the bottom you are seeing hide option. Will you please click that? Yeah, you can continue, Dr. Pramod. Go ahead. Everything is fine now.
Yeah. So to start with the history of the pharmacovigilance, as we all know, it has started a long back in the year 1962, initially with the United States of America. So as we all know, it is because of the thalidomide disaster that has brought up this concept called pharmacovigilance. So because of the introduction of this thalidomide product into the market in the year 1957, so it was basically started for all the pregnant women who are having the morning sickness. So during which, after a period of 3 to 4 years, all the scientists have observed that there is a birth of all seedling babies that is called phocomelia. In addition, the babies who are born to the mothers who took thalidomide are also having so many congenital heart diseases as well as many renal disorders. So, but in the United States, there was one scientist or physician who was a pharmacologist by profession. Her name is Kelsey. So that Francis Kelsey has not allowed that product to come into the US market. So because of which the United States could prevent all such phocomelia in all the young populations. Later, she was conferred with highest civilian award by President John F. Kennedy. So this is the brief history of this, how the pharmacovigilance has started. And soon after which, in the 1964, the UK has adopted the yellow card system. Later on, all the developed part of the countries has started this system of pharmacovigilance, wherein in the year 1968, the WHO has launched the program of international drug monitoring. But when it comes to the developing countries, it was very late. We could see this in infancy stage, and countries like in India, it was started only from the years of 2000s. So there was a lot of lacuna in this domain, wherein the safety of the products which are going to be get marketed are not completely monitored in all these developing countries. So if you see what are these adverse drug reactions, as we all know, the modern medicines have completely changed the way of the disease treatment. In spite of this, the adverse drug reaction still continues to be a menace which can be preventable. So there are a lot of studies that has been published recently, wherein they state that almost 40% of the adverse drug reactions that are occurring in the population can be completely preventable. And also it can cause further illness, disability, and death to the population. ADRs rank among top 10 causes of mortality globally. So because if we take that entire list of the reasons with which the populations are going to die, in which the ADRs are standing up in top 10 reasons. In addition to the disease conditions like cancer, HIV, and other serious heart diseases, these adverse drug reactions are also contributing very much for the mortality rates in entire globe. Because the significant factor that we can consider is main thing is polypharmacy. Nowadays, if you see any patient who is visiting any of the hospitals, they are not sent with only one drug. Majority of the times we could see there are plenty of the drugs that will be administered to the patient, even in the hospital setup or in the outpatient units. So this polypharmacy is a significant factor that is considered to be the rise in the adverse drug reactions. So this is one study which is published a decade ago, which I wanted to show you that adverse drug reactions and the risk factors among Indian ambulatory elderly patients. So this is the article which has been taken from Indian Journal of Medicine. So wherein you can see that what are the all risk factors that are associated with the adverse drug reactions. So the first thing that we could see is the age. So all the elderly patients, and again we have the same reason, elderly patients will have not only one disease condition, they will be having 2 to 3 comorbid conditions. So because of these 2 to 3 comorbid conditions, they will be prescribed with 5 to 6 medications, which again falls into the polypharmacy. And in this table, you can see the number of medications increases, the adverse drug reactions are also getting increased, which are also proved to be statistically significant. So in order to say that if you consider any drug to be safe, the answer will be no. There are no drugs which are said to be completely safe. There are risks in any intrusion that is done to the human bodies, whether it is chemical or surgical. So whatever chemical moiety that you are going to introduce into the human body, it is always associated with some or the other adverse drug reactions or the side effects. So nothing in this field is entirely predictable, except that the interaction between science and human may produce us surprises. So why I am quoting this sentence here is, so there are some adverse drug reactions which you will not be seeing in all kind of populations. Only in some populations who are having some gene mutation, you'll be seeing some particular kind of adverse drug reactions, which I will be showing in my further slides. And also in few cases, some adverse drug reactions will be occurring as per the gender specific. So you'll be seeing more adverse drug reactions occurring in the female population compared to the male population. So it all depends on various pharmacokinetic and pharmacodynamic parameters which influences the occurrence or production of these adverse drug reactions or the side effects. So that's why whenever we are dealing with any type of drug, we need to be very cautious. As I have mentioned that no product is a completely safe. So there is a need for strict vigilance on all the patients who are receiving any type of medications. Even if it is an established medication like paracetamol, we need to keep monitoring. So here you will get doubt because paracetamol is 100 years old drug. What is the need for doing the pharmacovigilance activities for these products? But in order to say that the new products are coming, we don't know which drug or which class of medication will have the drug interaction with the paracetamol. So because as we see, there are new type of drug classes that are introduced into the market. So the patient will be taking all these medications. Hence, we need to look for any kind of drug interactions that may occur. And also with the introduction of new food products, there is also chances of drug and food interactions. So in order to address all these things, there is a lot of need for looking into the safety of the patients, because at the end, whoever is the healthcare professional, it can be pharmacist, nursing, or a physician staff, we all look for the safety of our public and safety of our patients. So that's why first thing we need to keep in mind whenever we are going to deal with any product is the safety of the patients. So this is a flowchart how the pharmacovigilance is working currently with India from the government side. So if you see, basically it is divided into zonal offices, as we have 4 zones majorly distributed. North is at Ghaziabad, South zone we have at Chennai, West zone it is with Mumbai, and East zone we have with Kolkata. So all these zonal offices will further have so many adverse drug monitoring centers in individual hospitals. For example, if we take combined Andhra Pradesh and Telangana, around 25 adverse drug reaction monitoring centers we have. So if any patient wants to report any side effect or adverse drug reaction, they can report to these adverse drug reaction monitoring centers. These adverse drug reaction monitoring centers will be reporting it to the zonal offices, or they can directly report it to the national center. So once it is submitted to the national coordination center, that is at Ghaziabad, so they will be having some divisions. So first it will be checked with the quality review team whether the submitted reports are proper or not. Once the submitted report is proper, it will be sent to the next workflow. So wherein the higher committee will be looking into signal reviews, and the working group and steering committee will take further decisions on increasing the number of centers as well, and whatever the decisions has to be taken, it will be sent to the drug regulatory body, that is Central Drug Standard Control Organization, which is in the FDA Bhavan at New Delhi. So what are the recommendations that the National Coordination Center wants to give? It will be sent to the drug regulatory body, because at the end, these are the higher authorities which can take any decision on the drugs. So then the Central Drug Standard Control Organization will communicate it with all the marketing authorization holders that are present in India. So if there is any need for change in the package inserts to all the manufacturing authorization holders, that information will be passed on by the National Coordination Center of PVPA in association with the Central Drug Standard Control Organization. Then only this will be communicated to the WHO Drug Monitoring Center, that is UMC, that is called Uppsala Monitoring Center, present at Sweden. So this is the basic work chart, how it reaches to the WHO program. So basically you can remember it starts with an adverse drug reaction monitoring center, that is any hospital that is recognized by the pharmacovigilance program of India. So you may get doubt why so many adverse drug reactions that are happening in our country. So it is not only in our country. I will be projecting this next slides, the reports that has been reported to the national center, and you can see how the number has increased over the periods. So why so many areas are occurring? First reason is because of large number of prescriptions. If you see some patients, they'll be going to multiple doctors. So they will be receiving number of prescriptions. So with the increase in the number of prescriptions, the number of drugs that they are going to take will increase, and finally it will lead to the adverse drug reactions due to polypharmacies. Next is ADRs increases exponentially with 4 or more medicines that I have already shown in the previous slide. And sometimes it is also because of irrational prescribing. If you see in the rural areas, because all the public cannot access to higher hospitals, they'll be going to some registered medical practitioner that we call it as RMPs, who may not be having proper skills of prescribing some of the higher class of antibiotics or any kind of drugs. So definitely there we can see irrational prescribing. So once there is an irrational prescribing, it is always associated with the so many of side effects. And then important one is lack of proper system to detect or monitor these adverse drug reactions. So you yourself can understand this condition, the last point, that is lack of proper system to detect or monitor the adverse drug reactions. So suppose in your 30 years journey, for example, I am considering your age as 30 or 20 years, you might have taken medication so many times. So at least once you might have uh, experienced any kind of side effects like minimum headache or dizziness, but at that time you will not be knowing whom to report. So that is called a lack of proper system to detect or monitor. So many of the adverse drug reactions that are occurring in our rural areas are completely unaddressed because the HCPs that who are working in the rural areas as well as public doesn't know where to report such kind of side effects. So this is the main thing that the Indian government is addressing nowadays in order to percolate this kind of reporting system to all the rural areas. So there are some limitations to pre-marketing safety studies, because all of you are mostly from the pharmacy background here today. So you will ask me that we are conducting the clinical trials, then what is the need for even monitoring after conducting these clinical trials, right? So even though we are conducting the clinical trials, it is limited to very animal models sometimes, because some of the drugs we cannot give to some humans, and in some cases there are also information about long-term adverse effects is completely missing, because clinical trials maximum will be conducting for a period of 2 to 3 years, but there are some long-term adverse drug reactions which occurs after 4 to 5 years of using the particular product. So the best example is like carcinomas, which is occurring because of long-term usage of some of the medicinal products. So these long-term adverse drug reactions we cannot properly monitor during the clinical trials. And also I have quoted in the previous slide, there is very limited information about the drug-drug and drug-food interactions, because in the clinical trial the population you take will not exceed more than 5 to 10,000. So the food intake the enrolled clinical patients will be taking is not very diverse. But once the drug is introduced into the market, there is a very diversification of the use of this product in different regions as per the food habits also. So that's why that is an important limitation. Next is, in any clinical trial will not be enrolling the severely ill disease of who are having any liver or kidney diseases. So majority of the times in many of the clinical trials will be excluding them. So the information in these patients will be not present in clinical trials data. Hence, there is a lot of need in order to address all these things, and we consider all this as a major limitations of preclinical studies. So next interesting thing is what does this pharmacovigilance includes? So pharmacovigilance is not restricted only to the adverse drug reactions or reporting the side effects. The scope of pharmacovigilance has extended very well. Now we are also including all the type of medication errors. Again, medication errors, we have a lot of classification. Next is lack of efficacy. So when I was with Pharmacovigilance Program of India, we had addressed many cases of this lack of efficacy. So especially there is a good reporting that is done at Hyderabad and Tirupati. So wherein we have received lot of lack of efficacy cases. Means one example that I want to quote here is, so there was one adverse drug reaction monitoring center. They have reported us that patients were given with anesthetic, but they were not getting proper effect with that anesthesia. So that was initially considered because patients might not got enough dose with 1-2 reports. We can't judge completely on the lack of efficacy. So we have waited for a period of 1 month. Then the number has been progressively increasing. Then we understood that with that particular medicinal product, that anesthetic effect is not shown, and all the surgeons who are working in that hospital has complained the same reason. Whenever they are giving that anesthesia, patients are still experiencing a lot of pain. So then it was identified that it is not having proper efficacy. So like that, this lack of efficacy cases can be reported to the Pharmacovigilance Program of India. In addition, we have things called drug abuse and drug misuses. Majority of the times we deal with narcotic drugs and other things. Though we have so much restriction on the usage of this with all the pharmacies, still at many places or like eastern part of the world on our country, we do see this type of drug abuse cases and misuses. Of course, drug interactions with having a lot of workforce of young pharmacy students in our country, there is a lot of monitoring that is happening with terms of drug interactions and medication errors. So all this has been reported to the Pharmacovigilance Program of India. So as I have mentioned, there are a lot of medication errors that we deal with. To name few, we have like prescribing errors, which arises from the physician side. So while prescribing the medications to the patient, the physician can do some kind of errors. So those will be considered in the prescribing errors. So this prescribing errors is not that wrong product also. It may be sometimes wrong dosage or wrong formulation, any kind of thing. Whatever it is done from the physician side in the prescription, that will be considered as prescribing errors. So same way, we have so many things like dispensing, which can arise from the nursing staff, and preparation errors. Majority of the times during the compounding of the medications, we see this comes from the pharmacist, because some of the onco products and all still we need to prepare as per the body surface area, and then we are going to give the drug to the patients. So many a times these preparation errors are still occurring from the pharmacist side, and there is a lot of scope for us to monitor in all these parameters. Administration, again, as I said, whenever the medicinal product is administered to the patient by any physician or the nurse, the error can happen. Best example again here is use of inhalers. So majority of the times the asthmatic patients or patients who are suffering with chronic obstructive pulmonary diseases, they need to take these inhalers. So when the administration technique is not understood by them completely, we can see this type of errors. So once there is an administration error, the correct dosage will not be entering into the patient's body. So the proper desired therapeutic effect is not shown. So which also falls under the pharmacovigilance activities. So likewise, we have so many things which can be included in the medication errors class. So this one, you can see and understand how the numbers are increasing with number of years. So when initially the Pharmacovigilance of Program of India was introduced into the Indian Pharmacopoeia Commission in the year 2011, the first year we have reported out to 15,000 reports. But if we see in that year 2018, so it is reached almost to 80,000 reports. So you can only understand here what is the progress that has been done in the area of pharmacovigilance by the government with the help of all the adverse drug reaction monitoring centers. As the number of monitoring centers are increasing, as such, the number of reports are also getting increased. So which strongly recommends that there is a lot of scope that we need to bring all the MCA recognized hospitals into this program so that we can have complete safety around our country. And also, if we see the last 2 years data, the trend was almost same. And in the year 2020 and current year, because of the pandemic, there was less reporting as the focus was entirely shifted to the COVID. And if you see the age-wise distribution of the individual case safety reports that are reported to the Pharmacovigilance Program of India, the majority were reported in the age group of 18 to 44 years. but still a good number of reports that we can see in the elderly patients as well as in the pediatric populations. So sometimes when consumers are reporting, we will be missing some kind of data even if we do proper follow-ups. So that's why you can see in this slide unknown are around 7%. So you'll be getting doubt what is the quality that the reports will be getting in the national program. So this is the graph we can see. So the green line denotes the all other countries who are participating in the WHO monitoring program, and the blue line denotes the India. So you can see always the score of the individual case safety report is above 0.75 out of 1. Whereas if you see the green line, it is fluctuating from 0.3 to 0.6. So definitely the reports that are collected by Indian government are of quality, and it has a good data with the help of all the physicians that are participating in this program. So these charts will be available in the public domain given by the Uppsala Monitoring 7. So this Uppsala Monitoring Center will be giving it out for every quarter if you want to screen or year-wise. So because many of the pharmacy graduates are very much interested in looking up this adverse drug interactions. So if you see these drug-drug interactions, as I said, it is a potential cause for occurrence of all the side effects. It also accounts for 6 to 30% of all the adverse drug reactions. Suppose you are working in one hospital and you have reported 100 reports for that particular month. If you see in that 100 reports, 30 are arising because of these drug-drug interactions. So this is also taken from one of the Indian study in which they have quoted that the range is from 6 to 30%. And the frequency of potential drug-drug interactions among them, we can see is 4 to 46%. So hospital admissions due to drug-drug interactions are 0.1 to 2.6%. So in these hospital admissions, the patients are not getting admitted because of any disease condition. They are getting admitted because of the adverse drug reaction that they have experienced because of these drug-drug interactions. And also there is a lot of changes in pharmacokinetic and pharmacodynamic parameters because one drug will completely alter the action of the another drug. So because of that, the kinetic parameters present in the body also gets changes. So these are the few examples that you come across on daily use. So the best one is about usage of warfarin and enoxaparin, which increases the chances of bleeding in any patients. Next we have is simultaneous use of cyclosporin and simvastatin, which also leads to rhabdomyolysis, muscle tear we call it as. And one more example in the common activities or daily use is best example is aspirin along with clopidogrel can increase the bleeding. And if you see in the psychiatric departments, the usage of sertraline along with olanzapine will improve or increase the weight gain of the patients. And also there are some serious reactions that we encounter in usage of warfarin and doxorubicin, cyclosporin and doxorubicin, aspirin, clopidogrel, as such, we can say olanzapine and doxorubicin that will cause irregular heartbeats. So these are few examples to quote which we see in majority of the patients in the hospital setups. So once we get a proper data on any of the medicinal product, so by conducting the risk and benefit analysis, we are going to ban that product. And also because of the drug-drug interactions, some of the medicinal products that are banned here are, so the fixed dose combinations of iron with strychnine, arsenic and yohimbine, all these are the fixed dose combinations that are previously available with Indian pharmaceutical manufacturers, but later all these were banned because the physicians has identified that on usage of these fixed dose combinations, there are increase in the number of adverse drug reactions. So now we have strictly monitored that, and in the recent gazette that you have seen in the year 2019, almost 345 combination of medicinal products has been banned by the Indian drug regulator. And to quote few, fixed dose combinations of any other tetracyclines with vitamin C or concomitant uses should be avoided. Fixed dose combinations of antihistaminics with antidiarrheals, fixed dose combinations of laxatives or antispasmodic drugs in enzyme preparations. So all this such kind of combinations has to be completely banned in the usage of the public. So it is the duty of the pharmacist in the hospitals or in the community level. So whenever we are seeing any prescription with such kind, so we should strictly warn the patient to consult his or her physician and to address such kind of issues. So pharmacovigilance is not only restricted, as I said initially, to only monitor and report the side effects or medication errors or any type of lack of efficacies. Now, the recent trend in the pharmacovigilance is studies of pharmacoepidemiology, pharmacogenomics, toxicogenomics, and pharmacoeconomics. So in place of all this, the government gets so much benefit in designing the so many protocols as well as the schemes that they are going to introduce for the Indian population. So the best example that you can see in the predictive pharmacovigilance is the occurrence of Stevens-Johnson syndrome, which is considered to be fatal condition and which is a very serious adverse drug reaction. So there is a strong association between the usage of carbamazepine and experiencing Stevens-Johnson syndrome. But if you see, it is not occurring in all the patients, only in the patients who are having the presence of HLA-B1 502 allele mutation. So in that patients, we can see this kind of Stevens-Johnson syndrome. So this is called predictive pharmacovigilance, wherein, as I said in the previous slide, like translation pharmacovigilance, we can introduce such concepts of pharmacogenomics and toxicogenomics. So these two are entirely different topics which needs a separate session to deal with. So just in order to understand what is a predictive pharmacovigilance means. So before giving the medicinal product to the patient, initially only we can screen whether there is a presence of certain kind of alleles or certain kind of mutations in the patient, so that we can avoid giving that type of medicinal product to that patients. Hence, we can have the safety to the patient. So likewise, there are a lot of genetic variations that occurs in cytochrome 2C9 and VKO or C1 gene, which is associated with the altered anticoagulation response with the use of warfarin. So there is a big study that has been done on this warfarin studies. And also if you see in the developed parts of the world, there will be some registries called warfarin registry. Means any patients who are giving with warfarin will be entered into that register and they'll be completely followed up. So on following this type of information we can get, so that it will be helpful for us to prevent any kind of further side effects occurring in other populations who are going to take this type of medications. So this is the main use of understanding the adverse drug reactions and implementation of the same concept in preventing the adverse drug reactions. So majority of the students ask us this type of questions. Yes, we have identified some adverse drug reactions with some medicinal product X. So we don't know where to report on that. So if you want to report this directly, you can definitely report to the manufacturer, because any medication will have the package insert and the complete address of the manufacturer. So all the manufacturers will be sharing their toll-free number that obviously you can give to all the manufacturers. In addition, this data should be brought into the notice of the government of India. That's why we have this program called Pharmacovigilance Program of India. So all of you can go and visit www.ipc.com website we have. So in this Indian Pharmacopoeia Commission website, there is a section called Pharmacovigilance Program of India. So once you can access this website, you will get all the details. What is the toll-free number? What is the email ID? And what are the fax numbers that you want to share the data? All will be present in that And also not only for the National Coordination Center, the entire list of all the regional centers are also will be available in this website. So there will be no confusion in understanding where to report once you visit this website. And when to report, the moment you see any kind of adverse drug reaction, try to report that immediately. If not, advise to report that within a period of 1 month. So if you address that within a period of 1 month, the proper regulatory actions can be done by the drug inspectors who are present in that region. How to report? As I said, you can have multiple things. Email is there, telephone. So we do also have the mobile app only available for the Android users. I think for iOS and Apple users, they are still developing. So whoever is using Android mobiles, you can just go and visit the reporting app, which is developed by the NCC PVPI. What to report? As I said, all the reports you need to report. It is not that you will be reporting only the serious reactions. Even non-serious reactions, listed or unlisted, all the reports you need to report. So what is meant by listed and unlisted means, once you see any of the SMPCs or the package inserts that are given along with the medicinal products, they will be mentioning that on receiving this medication, some side effects are expected. So those are called listed. So it is not that only listed side effects that the patients will be experiencing. Even the unlisted reports also patients get experience. So in such conditions, all the listed and unlisted reports you need to report to the government of India. Who can report? Yes, anyone can report the adverse drug reaction. It is not only the nurse, physician, or the pharmacist. Even the consumers can report, or the relatives of consumers who are not a healthcare professional can also report this. So now you will get doubt how the consumers can report, because they are not trained or they don't know what information to be reported, right? So in the next slide, I will show how they can report that. Why to report? As I said, there are a lot of advantages that are present for doing this pharmacovigilance activities, because even from our side, we don't know when we'll be taking some medication. So we also can be prone to receive some medication in our future. So once we have established the safety, it is indirectly helping us to future. So this pharmacovigilance program of India has partnered with so many important stakeholders. So to name the important stakeholders, that first one is NTEP, that we call it as National Tuberculosis Elimination Program. So which actively do surveillance for all the anti-tubercular medications. Like majority of pharmacy students, as you know, there are different class of anti-tubercular medications. So even for the recently introduced molecules, this NTP will be accessing and doing the surveillance for the monitoring of all the adverse drug reactions. In addition, it is partnered with Universal Immunization Program, that we call it as UIP, which completely surveys about the adverse events following immunization. Because the immunization is very vast in our countries like India, wherein the healthy populations are receiving the vaccinations like polio and other things. So we need to also monitor the children who are receiving these vaccinations. Hence, PVA has partnered with this Universal Immunization Program. Next is National Vector-Borne Disease Control Program. So this particular body deals with the things like malaria, dengue, filariasis, and all the vector-borne related disease conditions. So they will be completely monitoring into that aspect. And final thing, initial first establishment was with the NACO, that is National AIDS Control Organizations. So all these are participating as a key stakeholders in reporting the adverse drug reactions to the pharmacovigilance program of India. So this is the details of the drugs which are withdrawn from the market because of the adverse drug reactions. So as I said, it is not easy for any drug regulatory body to simply ban any drug. there is a lot of expenditure that has been involved, and there is a lot of struggle that has been taken by the scientists to invent any new molecule. So because of occurrence of adverse drug reactions, we cannot simply ban that. But yes, at the same time, we need to ensure the safety of the population. So always we should outweigh the benefits are more than the risk. If the risks are more, definitely we are going to stop the usage of that particular drug. So if you see any websites that drugs are withdrawn from their countries, you can identify that terfenadine has been withdrawn because of the occurrence of ventricular arrhythmias, stilbestrol because it is known to cause carcinogenesis after the introduction into the market. Practolol is causing oculomycocutaneous syndromes. Nimesulide, which we are all using irrationally, it is causing liver damage, so it has been banned, but yes, only in the pediatric population. Adults can use this nimesulide. Servistatin, it is causing rhabdomyolysis in higher amounts, so that we have withdrawn. And COX-2 inhibitors, as we all know, it causes severe cerebrovascular and cardiovascular events. It has been withdrawn from the Indian market. And also, this list is a long. Just I have quoted few of the important drugs and the reasons for which these drugs were withdrawn. And you can see this pioglitazone use and the risk of bladder cancer. So it is not that only these adverse drug reactions has to occur in Indian population, then we take the decision. Whenever the information is reported in the neighboring countries, we need to adopt that and we have to immediately try to ban that in our country. So same thing was applied by the Indian government because pioglitazone was identified to cause the risk of bladder cancer. In the European region, so many countries has banned the usage of this pioglitazone. So by seeing this foreign data, Indian government has banned this drug initially, but later all the manufacturers has put a case that because no Indian has suffered with any bladder cancer, why to stop this in the Indian country? So then again, the government has revoked that. So then now again, it is introduced into the market and it is in use. So like us, who are us working for the pharmacovigilance, if we see the any retrospective data and if we can attribute any bladder cancer cases to pioglitazone, immediately we can report it to the government so that definitely they can take necessary actions. Because of the lack of data that has been still used in this country. So that's why there are sometimes some issues that will be occurring with the government and the manufacturers. So always it is advised that because always the manufacturers also looks for the safety information. It is not that simply they will be introducing into the market. They also definitely need some data to show that it has been reported to cause certain kind of events which is not good for our population. Then they will take the necessary action with some risk minimization plans or risk mitigation plans. So there are some very good teams that are working for all the manufacturers who completely works on the risk mitigation plans. So before in hand, if they have the data, definitely they will look into the aspects how to stop the usage of these products and safeguard our population. So it is not limited only to the age or gender or some of the molecules or the polypharmacy. It is also associated with race because if you see few of the adverse drug reaction, we can see in particular race or some countries like African countries. And also there are some new concepts called drug and herb interactions. Nowadays, the recently introduced things are hemovigilance, which is looking for the monitoring of the blood products. And also we have material-vigilance, which are working for the medical devices. Like the same way we can also have the herbal vigilance and also cosmetovigilance. So the trend has been shifting from the domain to domain. So whatever is the reactions that you are seeing with the Ayurvedic preparations and all, definitely we can report it to the concerned body. So there is a body that is present Ayush. We can also report to the adverse drug reactions that are occurring with the Ayurvedic preparations. So if someone wants to get more information on the pharmacovigilance, go to YouTube and you have this channel called Uppsala Monitoring Center. So they have posted so many videos in each area like risk management regulations, pharmacoepidemiology, and all the other things like signal detection and all. Or also you have basic of the pharmacovigilance courses. So all this you can access in the YouTube channels. Like then we can have in the information sources. So the next thing is journals of special interest. So as a student, if you want to have access to the journals which are specific to the adverse drug reactions or any of the drug safety interesting things, then you have these kind of journals like pharmacoepidemiology and drug safety or drug safety by ADIS, risk and safety in medicine, cases journal, and journal of medical case reports. These are few of the top journals that are of special interest. So we do also have a plenty of list that I can share with the organizers so that they can share with all the registered members to have the access. And we do also have the reference books. So if someone wants to understand the complete concept of this adverse drug reaction, we have some reference books. So to name few again, Myler's side effects, we have from Elsevier, we have Martindale, and we have pharmacovigilance from A to Z by Barton. So these are the famous reference books that any of the adverse drug reaction monitoring centers or the national centers will have in their library portfolio to quick review. And now it is not only limited to only few of the resources, because nowadays it is not only restricted to only particular kind of primary resources. We do also have secondary resources and tertiary resources. So when it comes to all these things, we have computerized literature screening process. So the famous one, which all the graduates know, is the PubMed. So yes, we have this PubMed, which is free to access, which doesn't require any login details for getting all the full text and all. And also you have some databases like Embase, EMC, famously we call it as Electronics Medicines Compendium, and current content and reactions weekly. These are online available. So whoever wants to have any safety data or published articles for that particular medicinal product, you just give your medicinal product in the search string and you can have that entire list of articles that are published in these databases. So the more you read the articles in these databases, the better you will be understanding the concept of the adverse drug reactions and the association. So in addition, we also have the national ADR bulletins. So for India, obviously we have the newsletters that is published by the Pharmacovigilance Program of India. In addition, if you want to look into the bulletins of the other countries, you can definitely have look into those websites. So the famous one, again, I have mentioned in this slide. For the New Zealand, we have it as prescriber update. For Australia, we have Australian Adverse Drug Reactions Bulletin. For Sweden, it is an information from Lac Media Certificate. For Canada, it is Canadian Adverse Drug Reactions. Finland, Taboo. Ireland, it is Drug Safety Newsletter, like that. So for each country, they'll be publishing some bulletins. It is available in their websites. Definitely, you can have a free access of all these newsletters. So when it comes to India, as I said, we have the reporting form that is given by the Indian Pharmacopoeia Commission. The version keeps on revision. So you can see the current version. What is the current version that the Indian Pharmacopoeia Commission is using from the website? And the same you can download from the website and document any kind of side effect, and you can submit them via email or fax, whatever is comfortable for your purpose. So you can see here there are two multicolored forms, right? One is in red color and another is in blue color. The red color form is especially for the healthcare professionals, and the blue colored form is for the consumers. So the blue colored form is designed not only in English but also in Indian languages. So total 18 languages we have this blue color form. So which includes all our southern part of the country also, like we have in Telugu, Malayalam, Tamil and Marathi. All these languages we do have the blue color form. But yes, again the red color form is only for the healthcare professionals who can understand what information has to be put into this form. Blue color form is very simple. It will have very basic questions like gender, age, and what medication you have taken and what has happened. That's it. But if you see in the red colored form, we need to give so many other details. So in order to quickly review that, I will not go into complete details because of the time constraint. So in order to project the main divisions of the red colored form, you can have these sections called patient information, suspected adverse drug reaction column, suspected medications, and reported details. See, these four are considered to be the mandatory fields, even for the industry as well as to the government, once you are going to give this information. Patient information, yes, we need to know what is the gender, age group and all, because based on that only we can take some regulatory actions. When it comes to the suspected adverse drug reaction, definitely we need to understand what is the reaction, what is the final diagnosis, because some of the symptoms will be grouped under one diagnosis. For one particular diagnosis term, we may have 5 to 6 symptoms. So in order to address all those things, we need to mention clearly what are all the suspected adverse drug reaction. And next we have suspected medications. It is not that only one drug will be involved in the reaction. So at that time when you are suspecting more than one drug, all the drug you can report that. That's why you can see in the bracket the S is mentioned. It can be a suspected medication or it can be suspected medications at a time together. And finally, yes, we do also require the reported details. Why we need to have reported details means once the regulatory body takes any action, it will be easy for us to communicate back. And we also can acknowledge the reporter who has addressed this or they have communicated to Government of India. So that's why these four fields are very mandatory and you need to consider whenever you are reporting any kind of adverse drug reaction. So what the patient information consists of, you can see here. Anyways, this red color form and blue color form also I will share with the organizers. So you can have the access. So these are all the details. So I want to focus here on the suspected adverse drug reaction. Compulsory always you try to note down what is the date of reaction started. It is not the date you have seen, but the date that patient has started experiencing the adverse drug reaction. Both are same. So to say an example, patient took some drug, consider aspirin, patient developed rash on 10th of July. But as a healthcare professional, 10th is a Sunday, so you have seen on 11th of July. But here you should not report 11th July. Date of reaction started means you have to always report as 10th July 2021. So that is the main difference. So many of the times when we see the students, they see to report when they have identified in the patient. It is not when you have seen, but you have to ask the patient when the patient started that adverse drug reaction, and that date we need to capture here. And down you can see describe reaction or problem. When you don't know what is the exact diagnosis of that reaction, definitely you can write entire description of that reaction. So once it is sent to the physician, they will analyze that. You no need to worry that you'll be not knowing what is the term that you have to use. Basically, you can describe that patient experienced rash so and so area on the thighs, hand or face, whatever it is. So entire description you can write. And when it comes to the suspected medications, you can see here, it is important to understand, you always try to capture the brand name or generic. And also you need to have manufacturers, if known, the company name, batch number or lot number, because each of the medication bottle or the strip that the patient is taking will be having this batch number or lot number. So once we identify this and we see any type of serious reactions, we can stop that lot or batch number of products. That's why we need to capture this lot number expiry date we need to capture, dose used, route used, because sometimes route will be misunderstood. That's why we also need to understand what is the route that they have used. Next, frequency, because sometimes by mistake once a dose can be confused with TID, like 3 times a day. So that's why all these informations are required when it comes to the suspected medications. And for what indication the patient has taken. Nowadays, if you see, there is a lot of off-label use. Means the drug is not introduced into the market for that condition, but still physicians use that based on their experience. So that is called off-label. So for what indication the patient has taken should be also be written here in the indication section. Finally, what you have is causality assessment. So causality assessment is nothing but you will be assessing whether this drug can cause this reaction in the patient or not. Basically, you will be assessing the causal relationship between the drug and the adverse drug reaction. So in order to perform this causal relationship or the causality assessment, we have almost 35 scales, but widely used are two scales, that is called WHO UMC scale or WHO UMC probability scale or Naranjo scale. These are the name of the scales. You can note down WHO UMC scale or Naranjo scale. These two scales are used in India also. So the same thing you can adopt for assessing this causality assessment. And the next thing, you can also report what is the suspected medication details and the other things. And concomitant medications. So concomitant medications is nothing but other medications that the patient has taken along with the suspected medications. Suppose patient is taking 5 medications and in that 5 only one drug is suspected for causing that adverse drug reaction. So in that case, other 4 drugs you need to write in the concomitant medication section. You should not think that other 4 medications are not of any use and no need to report that. Still, if they don't contribute to the adverse drug reaction also, we need to capture that in the concomitant medications columns. That's why this field is provided. Finally, reporter details. So the reporter is yourself, means you can write your name, your contact details, and what is your occupation. This comes under reporter details. So other important sections which are considered to be crucial for any report is like understanding of the relevant laboratory data. So whenever you are reporting any hepatitis case, always you need to try to report what are the elevated liver enzyme values. And whenever you are reporting the anemia, obviously you need to report the hemoglobin values. Likewise, whatever is the relevant lab data that is applicable to that adverse drug reaction, you need to capture that data. You should not leave any laboratory data unattended. All the information you need to collect and you have to capture in this form. Again, we have seriousness. So whether the patient died because of the adverse drug reaction or the mother has delivered any congenital anomaly in the newborn, sorry. So all this information you need to report. So if you see in few of the patients, there will be permanent disability because of the adverse drug reactions. So that also you can report. Yeah. So finally, if you understand what are the aims of this pharmacovigilance, first aim is to early detect of all the unknown safety problems. You can detect increasing in the frequency. So once you understand this increase in the frequency, based on the trend that is increasing, we can inform all the healthcare professionals. Identification of the risk factors. So once you analyze all the data that is obtained, we can do calculate or analyze the identification of the risk factors, which kind of population are more risk for this type of adverse drug reaction. Next is we can quantify the risk, obviously, and we can have rational and safe use of medicines, because there is a lot of irrational use nowadays, as I mentioned, in terms of not only for the antibiotics but also for the steroid use because of this pandemic. so that there are a lot of classification of the drugs which are irrationally used in Indian market. So in order to address all this, we need to compulsory have this type of monitoring system that is called as pharmacovigilance. So whatever data that India has obtained, the India will share that with the WHO. So once WHO has taken that data, they will be publishing in that WHO pharmaceutical newsletters. So I strictly recommend all of you who are in this presentation to just go and visit this WHO website, wherein you will get information on all the safety alerts from all the countries. So it is not only from India, though we are contributing to WHO. Likewise, all the member countries will be contributing, which are around 250 odd. So you can see all the information that is present. So this is one of the screenshot taken from the 2016 newsletter. So just to show that from very long back, we are doing that things. So here, if you see with the use of anti-rabies vaccine, we have identified the risk of erythema multiforme. So likewise, commonly used azithromycin, we have seen the risk of acute generalized exanthemus pustulosis. We call it as AGEP, because nowadays azithromycin is widely used. You can also keep monitoring for this reaction. So likewise, so much data has been shared by the Indian government with the WHO. So these are a few of the Pharmacovigilance Program of India recommendations to the Drug Regulatory Body of India. So you can see cefixime is causing AGEP that has been flagged as a signal and which was communicated in the year 2019. Same thing we have identified for lithium carbonate, that it is causing DRUS syndrome, that is called drug reaction with eosinophilia and systematic symptoms. And for a few of the drugs like chloroquine, which is caused to have toxic epidermal lacrinolysis TEN, and fluconazole for hyperpigmentation, these are asked for the manufacturers to update in their package inserts leaflets. So likewise, this information from the Pharmacovigilance Program of India will be shared to the drug regulatory body. Even though we have understood there is a lot of need for the pharmacovigilance, and we have understood what is the new trends, like what is the translation pharmacovigilance and all, and also we have seen what is the contribution that the Indian government is doing to the WHO as well as to the Indian public, still we have a lot of challenges and reasons why this is not full-fledged or why it is not completely taken into all parts of the country. So the first challenge that we have is lack of knowledge, because many of the healthcare professionals doesn't know that certain kind of system is existing in India. Next is awareness about the ADR reporting systems. So yes, they also don't know that where to report, when to report, and the other details. There is a lot of uncertainty regarding the causality relationship between the drug and the adverse event. So understanding this causality relationship is a very skillful task. So all the professionals cannot understand unless they get completely trained. But even though what we say from the government side or from the manufacturers per se, that is, the moment you see any adverse drug reaction, you have all the rights to report that. You no need to establish any causality, that you no need to confirm that this reaction might have occurred because of only this drug. Even though if you are not sure, always you are recommended to report that adverse drug reaction. Yes, we do also understand that causality relationship understanding is a very skillful task. Even though that challenge is there, still you are encouraged to report any kind of side effects. Next is lack of time or high workload. We need to also accept this, but definitely we need to overcome these kind of things. Whenever you see in any government hospitals, the healthcare professionals who are working are very less here. So that's why there is a lot of workload for them and they cannot contribute their complete time. Hence, this should be overcome by their respective hospitals by putting some strategies. Next is lack of understanding regarding importance of ADR reporting. Because many of the healthcare professional doesn't understand what is its important. They will ask us, what is the important to report this kind of report? So it is not that, even though if it is a known reaction, we need to always report so that we can understand what is the incidence rates. In order to have some pharmacoepidemiological data and also pharmacoeconomic studies, this will help the government to take some protocols or some actions or assigning some of the money in the budget. Once we understand the incidence only, we can do that. In order to get good budget to the pharmacovigilance system, we need to have certain kind of epidemiological studies and other things. Next is complacency, that they'll be satisfied with their working and also they don't believe that reporting is very important. So that should not be there with any of the healthcare professional. And the mostly that we have seen in the young healthcare professionals is about the lethargy or insecurity. So it is not that you are in low profession or nothing is like that. All the professions are considered to be equally important, and it is addressed by the health minister also. So you no need to feel any insecure when it comes to the pharmacist and physician thing. Always you can consult them for any doubt. Always there will be good physicians who can promote and encourage this kind of reporting systems. And as at graduate level, never we should be lethargic for doing such a kind of or certain type of decisions. And the last and foremost thing is about fear of medical legal consequences. It is very strictly mentioned by the drug regulator that there will be no medical legal consequences for reporting any side effects. Only if it is an irrational use, then maybe it will be addressed medical legally. If it is a prescription that has been given in a right dose to the right patient at the right time, there is no medical legal consequences that will come for any person who is reporting this adverse drug reactions. So in order to conclude my session, adverse drug reactions and PV remains a challenge in clinical and scientific discipline in our country. As you have seen, there are a lot of challenges I have mentioned in the previous slide. It continues to expand its domain due to ever increasing number of medicinal products, because you can see every 2 years or 3 years a new product is introduced into the Indian market. So there is a ever increasing trend of medicinal products. So it continues to expand and it is not a stop for any of the monitoring system. The benefits of drug therapy should always be greater than the risk. That is the concept or the idea that we should possess in order to extend these activities. Risk can be minimized by ensuring the good quality, safe, efficacious, and rational use of medicines. It is not that you are strictly monitoring, but regulators are looking for good quality of products and physicians will be looking for rational use. So once we work in an entire team, then we can achieve complete safety of Indian population. And the last point to conclude my session is that knowledge, education, and risk management strategies are crucial, are very important for minimizing adverse drug reaction. So it is always advised that all the working groups in any hospital to closely come together and share their knowledge with the government of India and with the manufacturers in order to have a healthy nation. So take home message, you can visit these websites. Prescribing, dispensing, and administration of drugs is not your responsibility, but taking safety and efficacy of drugs is your prime responsibility. So always you can join with all the social media platforms that young graduates are actively involved. You can follow them in Twitter, LinkedIn, and Facebooks. So you'll be getting the updates. So if someone get registered with them, they will also get some WhatsApp or text messages informations on the safety of the new products. So I recommend all of you to join the program of India so that it will be taken further to a good level. Thank you all.
Hello, Dr.
Thank you
Pramod.
so much, sir,
Excuse me,
for
Srujana
your
madam.
valuable
Hello, Dr. Pramod,
informative
will you
session
please
on
turn on your video?
pharmacovigilance and the vital role
Yes.
of pharmacovigilance team in the present scenario. And his session includes drug safety, adverse drug reactions, which were followed by the various divisions of pharmacovigilance team all over India, which is explained in the form of flowchart. And at the same time, he also included his session on duties of pharmacovigilance programmer and the various domains which are associated with it. And his lecture extended to drug adverse reactions, drug combinations which are banned, and concomitant medication, etc. Finally, Sir has given a wonderful explanation about the reporting form contents by means of various verbatic terms. Thank you, sir. So once again, I thank you on behalf of whole team of Department of Pharmacy, Chaitanya deemed to be University. Thank you so much.
Thank you, Mrs. Shrujna. And now, on behalf of Chaitanya deemed to be University, I would like to request the invited speaker, Dr. Pramod Kumar Garu, to receive a memento as a token of honor. Please, sir. Sir, please put on your microphone, sir. Your voice is not audible.
Questions also we can take up.
Sir, kindly put on your microphone on, sir. Yeah, thank
Yes,
you.
I'm doing that for Mangal. Yeah.
Thank you.
If any questions are there, you can write back to me. I can address that.
Yes. Thank you so much, sir. Now, I would like to request Dr. Chandana Garu, Faculty of Pharmacy, Chaitanya Deemed University, Warangal, India, to introduce the speaker of session 2. Over to Chandana.
Thank you, Vidya. Respected, distinguished guests, speaker, and friends.
Please unmute your mic, Dr. Chandana. Please unmute your mic, Dr. Chandana.
Unmute.
Thank you. Respected, distinguished guests, speakers, and friends. Good morning to one and all. It's a pleasure to extend my warm welcome to the speaker of session 2, Professor Dr. Satish Babu Natarajan, on the occasion of International Webinar on Emerging Trends in Pharmaceutical Sciences. Professor Natarajan has started his professional career in pharmacy and obtained his bachelor, master degree, and PhD in pharmaceutical sciences. He has published 23 articles in peer-reviewed international journals, and several manuscripts are in different stages of publication, and different research projects are going on. Sir has delivered 9 keynote lectures in many international conferences and universities. Professor Natarajan has received the prestigious Vice Chancellor Researcher of the Year Award in 2015. Sir has achieved several grants named Fundamental Research Grant Scheme on the topic entitled Nano Delivery of Papain for Colorectal Cancer in 2014, where the fund allocated was around 94,608 Malaysian ringgits. He has also received other research grants on catechin-loaded nanoparticles for the treatment of cancer and development of chickpea starch as a super disintegrant for fast-dissolving tablets. Sir has a patent on a composition for inhibiting proliferation of cancer cells by Intellectual Property Corporation of Malaysia. Professor Natarajan has more than a decade experience in the field of teaching, research, and industrial exposure. Professor has focused research interest in nanoformulation development, nutraceuticals, controlled and targeted drug delivery systems of cancer. And presently, Professor Natarajan is working as head, Department of Pharmaceutics, Faculty of Pharmacy, Lincoln University College, Malaysia. With this, now I request Sir to take over the session. And once again, I welcome you, Sir. Over to Professor Natarajan.
Thank you, Dr. Chandana, for your nice presentation. So before going to start my presentation, I would like to express my sincere thanks to Chaitanya University to provide this great opportunity to share my views on this entrepreneurial opportunity in herbal industries. So before going to begin, so I am going to express my sincere thanks to the chancellors, vice chancellors, registrar, principal, and head of the departments, faculty members, professors, lecturers, and finally the organizing teams and delegates who are all here. Okay. So now I am going to share. Can you able to see my slides?
Yeah, it's clear. It's clear, Dr. Satish.
Okay, sir. Thank you very much.
Yeah, you can go with your slideshow mode and you can continue.
Okay. Is it okay now, sir?
Yeah, please go with slideshow mode. It is still in thumbnail mode. Yeah, there.
Now you can see the slides, right?
Not yet. Have you pressed that from current slide option? Are you in slideshow mode at your end?
Yes.
But still we are unable to see. It is still in thumbnail mode only.
Oh.
Will you please reshare otherwise?
Yes, sir, I will reshare. I hope you can see the PowerPoint slide.
No. Yeah, Dr. Satish, it is still in thumbnail. For you, is it in slideshow mode?
Yes, sir. Yes.
Yeah. Through which mode you are sharing? Is it through window or entire screen?
Window, sir. Window.
Will you please go with the entire screen sharing otherwise?
Oh, okay.
Yeah, perfect. Will you please click that? Yeah, that's fine. Yeah, you can continue, Dr. Satish. Thank you.
Okay, sir. Thank you. Thank you very much. Okay, now I am going to present the wonderful topic for you. So I am not going to teach any subjective matters here. Right? So the topic which one I am going to discuss here is booming entrepreneurial opportunity in herbal industry. So before going to talk about the entrepreneurship in herbal industry, just I want to ask you a question from your side. Have you been dreaming of building your own business? If you want to become the leader, or if you want to become the boss, no need to report to your boss. If you want to become the leader, Or you want hustling in your side business before and after work, creating your empire on the low? Or are you ready for transition from employee to entrepreneur? If your answer is yes, yes. So this presentation is exclusively for you. Right? So can be called as entrepreneur? Entrepreneur is an individual who is able to perceive an opportunity for business and create an organization to develop and manage that business successfully. Those person is called as entrepreneur, successful entrepreneur. And moreover, he is able to identify and seize the opportunity, and sometimes need to create the opportunity and bring in necessary resources such as finance, workforce, and raw materials to develop and market the product and service which is required by the society or community. And he takes proper action that is imaginative, creative, and innovative. That's why they are different from the businessman. Businessmen normally they follow the traditional ways, traditional conventional methods, conventional policies and procedures. But whereas entrepreneur, they are willing to take the risk and to come up with the imaginative or innovative ideas. And a society that has entrepreneurs considered to be a successful society. So later slides, we are going to discuss why the society can be the successful society if the particular society have the entrepreneur. So now we are going to talk about the requirement, ideal features for the entrepreneur, those who want to become entrepreneur, how they are different from the normal employees. So employees wait for the right time to start their career, start their project, start their task. Whereas entrepreneurs, without thinking about the right time, there is no right time for that. They're ready to work 24/7. They won't feel the good day, Monday is a good day or Thursday is a good day. Employees aim for the promotion in the company. They are work hard people. They always work hard to get the promotion, to get the good pay from their boss, from the company. But whereas entrepreneurs aim for the promotion in the business. If the business is exploding, all the employees will be benefited. Right? So this is the mindset of entrepreneur. Employees always dream their plans, They want to implement, right? But whereas entrepreneurs plan their dreams, how the plan can be implemented. They will put into action. And employees think money is everything. They are working for money, get good pay. Whereas entrepreneur, time is everything. If you have more time, more than 24 hours, they can get more productivity. They can achieve greater productivity. By the way, they can generate the money. And employees always feel frustration. They are under frustration. Frustration is permanent for employees. But whereas entrepreneur, frustration is temporary. They know how to tackle it. They know how to escape from the frustrations. And employees always insecure about their freedom. But entrepreneurs free themselves from all their insecurities. Employees work hard people. Yes, of course, they are work hard people. Entrepreneur works smartly. Employees fall under particular job category. For example, particular person is working under as a production chemist. He's supposed to focus on the particular job category or job profile. They don't know about quality control, what they are doing. They don't know about quality assurance. They don't know about inventory management. But then in the case of entrepreneur, they create their own profile. They must be familiar with all the segments, all the departments in their company. And some employees blame others, especially when the project fails. But whereas entrepreneurs correct themselves, they find the mistake. What's the mistake? Then how the mistake can be rectified in the future project or the future operations. They try to correct themselves. Employees always look holidays as a relief. Yes. But entrepreneurs look holidays for strengthening their beliefs. What they will do in the weekends, Saturday, Sundays. According to this entrepreneur, they will do all the yoga classes, attend yoga classes, go for trekking, all the sports activities. So this is the mindset of entrepreneur. And importantly, employees break down after failure. Easily they give up. If the project fails, they give up very easily. But entrepreneurs wake up after failure immediately. Definitely, they won't give up. They won't give up. In the right hand side image, you can notice one of the picture. Purposely, I pasted here. It's a Jet Airways. That is Jet Airways. Most of you heard in 2018, the Jet Airways, the aviation company, shut down due to some financial crisis. But in 2021, this Jet Airways again tried to fly. They're preparing for fly again. So this is the mindset of entrepreneur. They didn't give up, right? So if you have this kind of qualities, the characters, features, definitely you will be the good entrepreneur or successful entrepreneur. Okay. If you don't have this kind of qualities, no need to worry. You can develop. If you have passion, if you have interest to be entrepreneur, if you don't have this kind of qualities, no need to worry. You can develop. You can inculcate yourself. It's a matter of your passion, matter of your interest. Right? Okay, fine. What is the status? What is the scenario, current scenario of the entrepreneur mindset in India especially? Very poor. Right? So around 1/3 of people, they become the entrepreneur when after 55 years old. 29% of people, they become entrepreneurs after 45 years old. And 24% of people become entrepreneur after 35. And only 12% of people, they become entrepreneur at the age of 25. Right? There is very less percentage, less ratio of people they are going towards to become the entrepreneurs. Because majority of the people, after the graduation, they are looking for the good job, MNC job, to get good pay. That's why the ratio is very, very low. Yeah, they don't want to take a risk. Right? That's what here the presentation is developed purposefully for you. So how we can change the trend. So what is the Indian students' mindset to become entrepreneurs? Why students are afraid of becoming entrepreneurs? Is it really afraid? So what the data is telling? Even with brilliant idea and correct marketing techniques, they hesitate to become an entrepreneur. A study conducted recently stated that young India was afraid to start their own business. Afraid to start their own business because they feel incapable due to lack of confidence and adequate knowledge required to start one's own business. That's a problem. So money is not a matter here. Right? The space is not better. The industry is not a matter. Only the mindset. Lack of confidence. That's why the ratio is very low, especially in India. So this is the mindset of Indian students. Okay, and what about the parents' mindset? So parents think that no one in their family have done business like this, and this is a game of risk. They don't have capital to apply. Thus, they discourage kids to do the entrepreneurship. Right? Investment. They think that need to invest more money. They need crores and crores to set up the business or to start the business. And second important factor is marriage. Who will marry a businessman? It's a big no in working class families, middle class families. The boy should be educated, well settled in the government job, and these days even they take software job, private software job or big MNC jobs. So these are the popular dialogues we used to hear from our homes, from our parents. Especially for girl entrepreneurs are much more. They feel it's not safe for single girl to do business. If she get married and then only do business, then something bad might happen, or they won't get through. So they want to face additional challenges, especially for good graduates. Right? So if you have a passion, if you want to be entrepreneur, right? If you have the qualities or you would like to develop your qualities, and moreover, still you need to try capable to convince your parents. So if you are capable to convince your parents and yourself, then you are the right person to be the entrepreneur. In order to be a successful entrepreneur, right, you have to start from small steps. The best way to achieve big goals is to take small steps. Right. Yes, that's what the government of India, they set up this organization, MSME, popularly abbreviated as MSME. What is that? It is a micro, small, and medium-sized enterprises developed by Indian government to boost this sector, micro or small scale industries. Already mentioned, no need to worry about the investment. It's a matter of your passion and interest. Right? Why the government of India is focusing these particular sectors, small scale industries? Because small manufacturing business can be started at your home. No need a big space. Big industry no need. You can start your business at your home without investment. Or you can start your business, industries, rental premises. Then what about machineries? No need. Sometimes no need, especially for herbal industry. Later I will explain you why machineries are not required. The cost of machineries or equipment required for starting small manufacturing business is very low. That's why the MSME is emerged as a highly vibrant and dynamic sector of the Indian economy. Why? Because these people, the small-scale manufacturing company, the unit, providing large employment opportunity at comparatively lower capital cost than large industries. You are not waiting for your opportunity, job opportunity. You are not looking for a placement. What you are doing? You are going to provide the job opportunity to your society. That's what the very beginning slide we mentioned. The society have the entrepreneur. That society considers a successful society. Why? Why? Because this aspect. Because you are providing the job opportunity to your society, your community peoples. And areas, thereby reducing regional imbalance, assuring more equitable distribution of national income and wealth. Very important point. This problem will arise. Equitable distribution, economically distribution. That's why the government of India set up this particular set of micro, small, and medium enterprises. The reasons. What are the reasons behind growth in MSME business? There are many reasons due to which the small scale business in India has witnessed its total growth. Small scale business. First one is high contribution to domestic production. You can increase the productivity. And no investment, already mentioned. The small investment. Few hundreds or few thousands you can start your business. Or zero investment. Without money, zero investment you can start your business. And significant export earnings. If you can meet the quality, in terms of quality, definitely your product can be exported to many countries so that you can generate money, good profit. Next, capacity to develop appropriate indigenous technology. According to your segment, the sector, you can adopt particular indigenous technology to enhance your productivity and operational flexibility. If you're the boss, you can decide what time you can start your business, when you can close your business, either morning or evening or 24 hours. So up to you, you can decide the operational flexibility. And technology-oriented industries. So you can combine or make a cluster with some other technology like artificial intelligence or IT sectors, right? So that the productivity will be enhanced and improved applications. If the domestic production is increased, obviously the import rate will be decreased. Location-based mobility. What is that? Location-based mobility. So today you can start your small business at Warangal. So later, after 1 month, you can go to other regional, other districts. Easily you can move from one place to another place. Very easily, very feasible. Why? Because we no need to shift big instruments, big machineries. Very easy. Easily we can shift from one place to another place. There is a chance of easy shifting, low intensity imports, and competitive in domestic and export market. So these are the reasons why the MSME business are growing very well. Okay, what are the sectors in India has more opportunity for entrepreneurs? So what are the sectors? So these are the areas: food product manufacturing, healthcare sectors, education, textile, energy sector, tourism, automobiles, and waste management and organic farming. So these are the popular sectors, have huge entrepreneurial opportunities, very huge. Okay, so since we are the pharmacy people, pharmacy graduates, pharmacy students, how we can start, how we can combine the entrepreneurial mindset into pharmaceutical sectors. So we are not popular with automobiles or tourism or textile or waste management. We are purely from pharmacy. So how our subjective or scientific skill can be converted into entrepreneurship mindset. Right. So the next slide, I am going to show you how you can combine it. Herbal entrepreneur. Herbalism is a unique niche for aspiring entrepreneur. Herbalist entrepreneur can create their dream job by turning a patient of herbalism into living. Why? Because they can create line of herbal extract, body care products, or tea blends, growing or while crafting herbals to sell, or creating monthly herbal CSA shares in unique recipes are just a few of the possibilities herbalists have been ventured into their career. Very easy. You are studying about the pharmacognosy, right? Pharmacognosy 1 and advanced pharmacognosy and natural products, right? Many modules. With that subjective skills, convert the subject skills into entrepreneurial mindset. You are doing many extraction in your practical sessions, right? So you export that particular extract to other countries, or you sell that extract to the market, you will get money. That's it. Very simple way. That's why unique niche. You will also be able to build a business with a positive social impact if you are a herbal entrepreneur. In the picture you can see how you can provide a job opportunity for your society or your communities. There are illiterate people, uneducated people. So one of the ladies drying the herbal leaves, right? Another one, some of the people they are sorting the flower petals. By the way, we can provide job opportunity for these type of people. Obviously, your end product will become part of someone's healthy lifestyles. But where you get your supplies and ingredients can make a difference. So it's very simple. Just you collect the leaves, do the extraction, yes, okay, fine. But you need to ensure the quality of your raw materials, where you are procuring, where you are purchasing. That matters, the quality matters. You need to ensure the quality. Why? Because the end product will going to meet someone a healthy lifestyle. So many small business owners work part-time on this side, part-time, until it grows enough that you can replace full-time work elsewhere. Or suppose you are working somewhere, the salary is not sufficient for you, still you want to make more money, right? So you can start this type of business as a part-time. Thankfully, herbal business are no different. They can be scaled to fit available resources and grow slowly over time. Definitely, yes, if there is a quality. If your product is meet the particular quality standard, definitely your business will grow faster. Okay, so what is the market potential of herbal products? So you are telling about, okay, you can be the entrepreneur, these are the qualities required, okay, you can build the product. Yes, okay, what is the market scenario, market potential for this particular industry, medical industry? See, the India is the second largest exporter of medicinal herbs in the world, second largest. India is second to China. China is dominating this field. India is second to China with total of more than 6,000 medicinal plants in the country. They both produce, India and China both produce up to 70% demand for herbal medicine over the world. See the market potential. And what is the percentage contribution to the export of medicinal aromatic plant product from India? How much amount of product we are exporting? Around 60% of people, products are exported in the form of crude herbs. What is crude herbs? Like you just source your first leaves, dry it, pack nicely, and you can export. Very easy. How much percentage? Contributed 60%. Around 30% of products are exported in the form of semi-finished products, and 10% exported in the form of finished products. So in terms of value, the export value, how much? In the year 2015, around 300 million US dollars. The export value is around 350 million US dollars. Where in the year of 2019, the volume is gradually increased up to 450 million US dollars. So means there is a huge potential for this herbal industry, especially for export. Now we are talking about only the export aspect. What is the global herbal medicine market revenue by its product? So already we discussed 3 ways we can export our product in the form of crude drugs, semi-finished products, and finished products. Okay. So what is the value of the particular products? In 2014, around 63 billion US dollars altogether in the tablets, capsules, powders or extracts, fruit leaves or seeds, bark, etc. So it is expected to reach in 2024. It is expected to reach more than 100 billion US dollars. That's a huge potential. So if you have interest to become the herbal entrepreneurship, you can tap this great opportunity. So medicinal plants, what is the scenario about medicinal plants in India? I already mentioned, India is the second largest exporter of herbal medicines to other world, next to China. So India has a 15 agroclimatic zones that comprise more than 80,000 types of plants, which of more than 6,000 have therapeutic medicinal values. So these medicinal plants are used in numerous applications in the Indian society and used to make medicines in traditional medicinal practice such as Ayurveda, Unani, Siddha, and homeopathy, also used in plant-based pharmaceutical companies. More than 960 types of medicinal plants are traded, of which 178 species have yearly consumption of more than 100 metric tons. The requirement is very huge. Around 80% of medicinal plants are extracted from wild, while 69% plants are collected using destructive farming practice. So means around 80% of the plants are collected from the wild, from the mountain, from the hills, from the forest. They are not cultivating. Just people said they go in there and collect the materials and properly sorting and ensure the quality and pack nicely and export to the countries. So what are the plants? Medicinal plants have high demand in India throughout the year. Number one is aloe vera, very famous herbal product. Aloe vera, good to licorice, medicinal soft medicine circle, ashwagandha. Ashwagandha is one of the best, one of the excellent medicinal herbal product. So later I will tell you why ashwagandha is, I stress very important. Ashwagandha, not only ashwagandha, and moreover lemongrass, chamomile, patchouli, tulsi, and palm also. These are the medicinal plants. High demand medicinal plants in India. So now I am going to explain about what is the importance medicinal plants in Telangana region. See what's it. So what's the impact of medicinal plants at the Telangana state? Telangana is the ideal terrain for medicinal plant cultivation. The dry and arid zones in Telangana can provide ideal terrain for cultivating some varieties of medicinal and aromatic plants. Many plants. So according to Dr. Utis Kumar, Central Institute of Medicinal Aromatic Plants, Telangana soil is suitable for cultivation of aromatic plants like lemongrass, almosa, and citronella. That's what I mentioned earlier. So these are the plants. These are the plants are identified as a high demand plants in India. Lemongrass. So where these plants are cultivated? In your state. Lemongrass, almosa, and citronella. So what they are doing from these plants? They are extracting oils from this plant, essential oils. So these oils extracted from this plant have both aromatic and medicinal values. These products have immense popularity and have a feasible market in India and abroad. What you can do here? So in the first picture, the top right hand side, you can see some of the people they are cultivating the herbal medicines from the field. You can cultivate the particular medicinal plants, or you can source this material from the farmers and you can do the extraction. You can set up the extraction unit at your place. In the second picture, you can notice one of the guy he is standing near to the distillation, steam distillation unit. Means you can collect the materials from other farmers, then do extraction, collect, you can make the oils and you can export it. Export to the country, or you can supply to the pharmaceutical company or cosmetic manufacturers. So apart from this dry region, the state block soil is also conducive for ashwagandha cultivation. Ashwagandha root. Ashwagandha is one of the more demanded product, high demanded product. Ashwagandha, which is famous for its pharmacological properties used for treatment of wide range of ailments. Right? So out of 10, nearly 4 to 5 medicinal plants are cultivating in Telangana state. Out of 10. The status of medicinal plants at Warangal, your district. Know about your district. What is the status? Warangal district is reputed in Telangana state reputed as a habitat of medicinal plants. So this hill is more popular for Swami Narasimha temple. Right? So it's more popular. You know very well, right? So here, this location, this hill is assigned as a medicinal plant conservation area, which covers nearly 200 hectares. This hill, you can easily notice this type of medicinal plants, herbal materials, which is called as a Navatampa, Chenglava Kustu, Chamakura, Neelatadi, Haridra, Pink Nodding Orchid, Kaliga Rastagada, Chukudampa, Mungri Mokla. So these are the medicines, herbal medicines available in this Mulro. If you have a chance, right, go and find these medicine range. Once you collect it, you can source it from there and you can do proper process, drying, packing. Then you can supply to the local market or the companies. Okay, now we are going to discuss about supply and demand. Already we discussed there is a huge potential for this herbal industry. It's growing gradually. So what about the supply versus demand scenario? There is a huge gap between the supply and demand of medicinal plants in medicines in India. According to All India Trade Survey of Prioritized Medicinal Plants 2019, demand for high-value medicinal plants increased by 50%, while the availability is declined by 26%. See the difference. The requirement is increased 50%, but the availability is declined by 26%. So we can have this opportunity. If you have interest to cultivate, go and cultivate. If you have strong network, your farmers, the local farmers, ask them to cultivate these type of medicinal plants. Easily you can sell to the market. Utilize this opportunity. So these need to increase habitat irrigations. And cultivation of medicinal plants in commercial mode is one of the most profitable agri-business for farmers in India. If anyone has sufficient learn and knowledge of herbal marketing, then they can earn high income with moderate investment. Cultivation of medicinal herbs such as Sangapushpi, Atis, Tooth, Turkey, Apikachu, and Pancha changing the Indian agrarian Ayurvedic scenes and providing extraordinary opportunity for farmers to increase their revenue. So according to the traditional treatment, health centers, there are 25 significant medicinal plants that are always in full demand. So already previously we have noticed there are 10 medicinal plants have a great demand in India all throughout the year. So apart from that, so some more medicinal ingredients like Indian barberry, licorice, circulatis, woodwood, chandan, senna, long pepper, dami, jatmalsi, madhunashini, kalmik, satavari, aswagandha, chirata, katti, sankushpi, ashwagandha, hukum, sakhi. So if you have a network with the farmers, you go and ask them to cultivate these materials, these medicinal herbs. They can get good income. Why there's a huge demand, the huge growth in this herbal industry? What are the reasons? According to the credible source, global herbal medicine market is expected to reach a valuation of $400 billion by the year of 2026. How much? $400 billion. Not a very small money. Why there is a huge increase? Because the product adaptation, the herbal product adaptation by the population of the world. Just we can link with the COVID-19. Many of the people, they consume many herbal products to get good immunity to fight against the COVID-19 virus. So good adaptation and rising support and funding from government for research and development of herbal medicines. These are one of the important parameters. Government is providing huge money for the R&D development of herbal medicines. And easy availability of product are the major factors propelling the industry growth. Herbal products are easily available. So one of the best examples is the Himalaya Wellness Store. So now you can see the wellness store, Himalaya Wellness Store in all the metros. Easy availability. And further benefits such as low cost and no side effects compared with allopathic medicines are also contributing to herbal medicine market outlook. And moreover, launch of current good manufacturing practice by the FDA or license agreement are expected to drive the significant growth. So these are the reasons. For these reasons, the herbal industry is growing nicely, gradually. Okay. Yes. Okay, sir, fine. There is a much potential. I want to be an entrepreneur. So you said herbal industry is good. Okay, sir, fine. Right. How to start? How to begin my entrepreneurship from this herbal industry? What I need to do? Right. Now you can see one of the most popular leaf. Right. You can recognize what is a leaf displayed here. This leaf is called the miracle leaf or miracle tree, monumental tree. So what is the leaf? Name of the leaf? This one. It's nothing. Moringa oleifera. Drumstick Moringa oleifera. This is called as miracle tree. Moringa oleifera is called as miracle tree and monumental tree. Why this is called as miracle tree? So later slide I will show you why this tree is called as miracle tree and monumental tree. Moringa oleifera from the Moringaceae family is a perennial tree widely cultivated in many tropical regions and easily grown in adverse conditions. It is used to treat several conditions such as malnutrition, diabetes, blindness, anemia, hypertension, stress, depression, skin diseases, arthritis, joints and kidney stone disorders, and so forth. And moreover, capacity to help in maintenance of cardiovascular system and have a powerful antioxidant, anti-inflammatory properties, used to treat many types of cancers, as well as regulation of urinary tract and lactation in anemia. Also been applied to cosmetic and byproduct due to high nutritive and protective properties of the oil. See this picture. So now we are going to compare this moringa dry leaf with the other food supplements. So the 100 gram of moringa dry leaf, just 100 gram of moringa dry leaf is compared with the different food substance or beverages. This moringa dry leaf contains more than 10 times of vitamin A than carrots. As we know that carrots is popular for its vitamin A, but moringa dry leaf have more than 10 times of vitamin A than carrots, and oft times vitamin C of orange. Orange is popular for its vitamin C. Moringa oleifera have more vitamin C than orange, and 17 times of calcium of milk. So it is popular for popular beverage contain more amount of calcium. So how much percentage? How much weight is greater in moringa oleifera? 17 times. 15 times of potassium than bananas. 20 times of iron of spinach. 9 times of protein of yogurt. That's why moringa oleifera is called as miracle tree, because it contains high nutritive power properties. That's why moringa oleifera is called as miracle tree. The latest slide I am going to explain why this moringa oleifera is called as money making tree. Okay, so how the moringa oleifera can helpful to become the entrepreneurship. Okay, so what we are going to do, we know raw heads like a fresh moringa oleifera. So if you sell the fresh moringa oleifera leaf into the market, the price is maybe 5 rupees per kg. So what we need to do here as a pharmacy graduate, if you want to become entrepreneur, so we have to convert the raw heads into value-added products so that the value of the product will be enhanced. By the way, we can get more profit. Right? So in order to convert into value-added product, so these are the process we need to be carried out. So what are the process? First, we have to collect the raw materials at the right season and right maturity. To get the right composition of phytochemicals or essential oils, the collection or harvesting should be carried out at the appropriate stages and seasons. This is very, very important. Okay. After collection, we need to grade and sort the materials. It's a basic procedure like grading and sorting or effective means of getting higher value of the produce with the minimal efforts. Next, cleaning. So cleaning should be followed to remove the unwanted foreign materials and pollutants. So depends upon the materials, herbal materials, nature of the materials, we need to use different cleaning procedures. Right? Next, drying. So depending upon the nature of the materials, we have to choose the appropriate drying techniques, especially sun drying or solar drying or shad drying or microwave. Different drying techniques are available. So we can pick either one of the drying technique according to the product. Packaging. Packaging is very, very important. It makes a more aesthetic appearance, and moreover it keeps the control from extraneous matter or from loss of material under normal conditions of handling, shipment or storage. Different categories of plant materials require different packaging consideration. Some material you can pack with the plastic TP bags, or you need to store in the glass bottles, glass containers, or barrels, or aluminum tins, or iron tins. Right? So you have plenty of packaging options there. So you have to choose according to the nature of the materials. So these are the common procedures, basic procedures you need to carry out for convert the raw goods into value-added products. Right? Okay. So how these moringa oleifera fresh leaves can be modified or converted into value-added products? What are the different forms we can be able to convert them? So the middle picture you can see the fresh moringa oleifera leaves, right? So you collect the fresh moringa oleifera leaf, right? Clean properly and dry it, right? You will get moringa oleifera dry leaf. That's it. This is a value-added product. Convert into dry leaf, put into the plastic bag, right? And seal it, and you can supply to the buyer. Very easiest way. Or you can convert the dry leaf into coarse powder, or fine powder. So you can convert it to moringa powder or coarse powder. Or you can collect the seeds, you source the seeds, moringa seeds, and convert into moringa seed oil. Where you can use moringa seed oil? Moringa seed oil is high demand. There is a high demand in cosmetic industry. Moringa seed oil. Do you know the price of moringa seed oil per liter, per kg? More than 1,000 rupees per kg of moringa seed oil. There is a huge market for this moringa seed oil. Right? And you can convert into moringa candies, moringa jellies, or you can add the moringa powder into the candies, the energy bar, peanut candies, or you can add into the soup base. You will get moringa soup, highly flavorful, nutritive soup. Otherwise, you take the moringa leaf, the coarse moringa leaf powder, put into the deep bag and seal it. You will get moringa deep tea. Right? There is huge market potential for moringa tea bags. Huge market potential. Do you know the price of one dip of moringa tea? One dip, just 2 grams of moringa oleifera. 2 grams. Just it contains only 2 grams of moringa. The price is 4 rupees. Just 2 grams of moringa oleifera. See the market value of this product. Another one is moringa energy bars. Or you can fill into, encapsulate into hard gelatin capsule. You can easily convert into capsule. Moringa oleifera capsule. Or you can fill into the jar, PET jars. Right? This moringa oleifera you can put into your teas, regular teas, or you can add into your foodstuffs to enhance the nutritive value of your food. Right? So not only for food and beverages, this moringa, already the moringa seed oil is used for making cosmetic products. Right? So next we are going to see the market value of the moringa seed. Apart from that food and beverage purpose, this moringa powder and seed oil can use for making soaps, moringa soap, or we can use prepare face creams or moringa shampoos. So these are the market potential for moringa products. Okay, sir, this is very easy. Yes, we can convert it into this value-added product from powder, dry leaf, coarse powder, or we can able to make energy bars and so forth. Okay, fine. So what is the value of this product? Easily we can make. What's the value? Product value. So here I explained that the leaves, the fresh leaf, the price of moringa fresh leaf is 10 rupees per kg. Just 10 rupees, maximum price. The same leaf, if dried, the value of the moringa leaf is up to 200 rupees. 1 kg of dried moringa leaf, 200 rupees. If you have a pulverizer in your lab or in your home, there is a blender in your home, mixie is there. If you can convert the dry leaf into powder, the price will slightly increase from 200 to 300 rupees. Or you can just clean the moringa seed, just you clean it, you sort it. The wings, you clean the wings and nicely pack. The price of moringa seed is costing around 1000 rupees per kg. If you can able to extrude, right? If you can able to extrude the seed, if you convert oil, the price is more than 1000 rupees. Energy bar, price of the energy bar, moringa energy bar is 50 rupees for 10 gram size, just 10 gram size, 50 rupees. The soup powder cost is 100 rupees for 100 gram powder. And tablet, what is the cost of the moringa leaf tablet? 1000 rupees. How many number of tablets? Just 20 to 30 number of tablets. What's in that tablet? Just your normal tablet excipients and only your moringa powder. That's it. Once you compress it, the product value is more than 1000 rupees. Similarly, the capsule, moringa capsule, it's also costing more than 1000 rupees for 20 number of capsules. So now you can understand the market potential, the value of this industry, herbal industries. So just here, discuss about only one particular product, moringa product, for your understanding. This is applicable for all type of herbals. It may be tulsi. You can apply this concept to your mint leaf, or ashwagandha root, or aloe vera, anything. What we are doing is just we are convert the raw herbs into value-added products. What we need here? We need small instruments like a mixie. The domestic mixie blender is sufficient to make a moringa leaf powder, or mint leaf powder, or tulsi leaf powder. Very simple. What about investment? No need. Just you can cultivate this moringa oleifera tree in your backyards, or what areas, or in your medicinal garden, your college premises, your university premises. Right? So these are the possibilities. See the market potential of this moringa product, only moringa product. Previously, we discussed about the overall medicinal product value, market value in India as well as overseas. In 2018, the market potential, market value of this moringa product is around $6 billion US. $6 billion US alone moringa. How many number of herbal medicines are available? More than 6,000. Right? So in 2025, it is expected to reach more than $10 billion US. $10 billion US. Right? Okay, fine. Then what's the role of academic institute? Right? To develop, to generate more entrepreneurs. So entrepreneurship process at university can make a very significant contribution to promoting meaningful entrepreneurship. Because we are teaching all the subjects, pharmaceuticals, pharmacy practice, hospital pharmacy, pharmacology, everything. But we are lacking in this area. So we need to start this type of activities, short courses about entrepreneurship, the opportunities of challenges, how to begin the marketing skills, everything. So entrepreneurship course at university can make a very significant contribution promoting meaningful entrepreneurship. And academies are able to alter the mindset of students and to have the ability to effectively teach basic business skills and develop creative thinking in their students by giving them practical assignments. Is convert the practical into project oriented, business oriented, so that they will gain scientific information as well as entrepreneurship or business skills. And when to have guest speakers and guest lecturers, that this tend to offer measure of interaction with the entrepreneurs, but it's not really enough. So the placement cell is very common to all the universities, all the institutions, but very rarely we can find these entrepreneurship cells in the particular institutions. So similar to placement cell, if we can able to develop, we can set up entrepreneurship cell in all institutions, all the universities or organizations. Definitely we can able to generate more entrepreneurs from outside, from our academic side. Government initiatives. How the government is boosting this herbal industry development. The National Medicinal Plan Board offers up to 75% subsidy to farmers. That's what I mentioned. Don't need to worry about investment. Government offers more loans, more schemes, right? And government schemes and guidance for financial assistance in various zones of medicinal plantation, secure under promotional and commercial plans which are relevant for the government and non-government facilities. Right? And the second one is the export promotion of several herbal products has been assigned Pharmaceutical Export Promotion Council, is popularly abbreviated as Pharmaceutical. This committee facilitated the exporting community and undertake various promotional measures to promote exports of their products. The pharmacists only focus for the promotion, export product promotions. And the Department of Commerce has set up export product councils to promote exports of various product groups and has assigned Shellac and Forest Product Export Promotion Council. So popularly abbreviated as FACTION, to mandate export of herbal and medicinal plants. So not only that, the popular Ayush Department, International Cooperation Scheme by Ministry of Ayush, provides financial assistance to exporters to help them participate in trade fairs, organize international business meets and conference, and avail product decision reimbursements. So no need to worry about that. Government also supporting for this particular industry. Right? And futures of medicinal plant industry in India. In India, the production and cultivation of medicinal plant is mostly unorganized. Don't feel this negative. If this is unorganized, if you are capable to convert this unorganized sector into organized way, proper systemic way, definitely there is a huge market in overseas especially. An equipped supply chain management and formation of farmer association will improve the production and sales of medicinal plants in the country. So better you tie up with your farmers, the local farmers. The sector has observed recently for sort of bringing the technology upgradation. Right? So these startups are using precise farming techniques by integrating artificial intelligence and data analysis for crop profiling, seed analysis for better germination, among others. So what are the opportunities? Growing, selling, and processing of medicinal and aromatic plants, good opportunity. Value addition is another great opportunity, that is value added products. It's fetch higher price than raw ingredients, raw materials. Contract farming is another opportunity in this sector. Most of the companies doing contract farming, like Himalaya or most of the pharmaceutical company, they are doing contract farming. Means they have the collaboration with the farmers, local farmers. In case of medicinal yam, for example, the medicinal yam company purchased dry yam at the rate of 1 rupees per kg, having 1% dieting. Means the product have 1% dieting, the price is 1 rupees. The same medicinal plant, the medicinal yam contain 3% of diacetyl, the price is 3 rupees. So according to the proportion of the phytoconstituent, the price is differ. The quality is increased, the price also increased. Right? Yes. So now it is to the conclusion. So what is the right time to become an entrepreneur? There is a never right time to be an entrepreneur. Honestly, there is no right time for anyone to become an entrepreneur. It's already mentioned, but first quality of the entrepreneur, there is no right time for entrepreneur. So always 24/7 there is right time. It could be happening in your 20s or in 60s. It's up to you, up to your mindset. You can be the entrepreneur by today itself, now itself. Someone can be entrepreneur after 5 years, after 10 years, maybe after your retirement. It's up to you. So if you want to become an entrepreneur, you can start now itself. Why? Because this is a COVID time. This is lockdown time. You have plenty of free time after your online lessons. You can go to your backyard, find what are the herbs available. You just pluck it, process it, dry it, pack and sell to your local traders. You will get money. No need to wait money to pay your tuition fees. Don't depend on your parents to pay your tuition fees. You can generate your money. At least you can generate pocket money for yourself. Right? Like neem tree. Easily you can find neem trees in your yards. Right? You just collect your neem leaf, dry it and sell. What is the price of dry neem leaf? 100 rupees per kg approximately. 100 rupees neem dry leaf. Neem leaf powder, 150 rupees, 200 rupees per kg. So if you want to become an entrepreneur without investment, without big industry, right? Without machineries, this is a ways. Plenty of ways. Right? If you are an entrepreneur, you can able to change this damaged road, the patchwork road into blossom road. If you are an entrepreneur, if you have the mindset to be the employee, you may use to follow others, follow your seniors. You used to follow like highways. Those who will choose this type of road, the damaged road, the entrepreneurs. Entrepreneurs choose this type of road to convert this road into blossom road. Right? I hope this presentation is enlighten your mindset to be the effective, successful entrepreneur. Wish you all the best.
Sir, thank you for your excellent talk. It's been very informative for all of us. Thank you for providing us the difference between an employee and an entrepreneur. In the present scenario, this transition from employer to entrepreneur of young pharmacists is an excellent shift. You have enlightened us with your guidance and guidelines to how to initiate a small scale business profoundly towards herbal or organic farming. Thank you for providing insights into market analysis and medicinal plants of Telangana, importantly Moringa, and how to transform raw herbs to value added products. I appreciate your ideology of entrepreneurship cell in academic institutions. We are glad to have you for the session, sir. I strongly believe that most of the participants have rightly connected to you with your vision and its scope. Thank you very much, sir. Over to Divya.
Thank you, Dr. Chandana. Now, on behalf of Chaitanya deemed to be University, I would like to request the invited speaker, Dr. Satish Babu Natarajan Garu, to receive the to receive a memento as a token of honor. Thank you so much, sir, for accepting our invitation in spite of your busy schedule.
Thank you, madam. Thank you for your valuable time and delegates. If there is any question, you can put to me or you can contact me. So I am available. I can ready to teach you how to be the way how you can start your business. Right? So I am always available to help you. Okay, thank you, madam, especially for the organizing committee to provide us great opportunity to deliver this wonderful topic to all the delegates. Thank you.
Thank you, sir. Now, I would like to request Mrs. Visalakshi Garu, Faculty of Pharmacy, Chaitanya deemed to be University, Warangal, India, to introduce the speaker of session 3. Over to Mrs. Visalakshi.
Thank you, ma'am. Now, it's my privilege to introduce our speaker, Dr. Vasudev Rao Garu, Aupati, Senior Lecturer, Department of Pharmaceutical Chemistry, E-Learning, Lead School of Pharmacy, International Medical University, Malaysia. Sir was awarded PhD in Pharmaceutical Sciences from Andhra University in 2013 under the supervisor of Y. Rajendra Prasad Garu, current professor and principal, Andhra University College of Pharmaceutical Sciences. Sir has secured prestigious CSIR Senior Research Fellowship from Government of India for completing his PhD degree. Sir is a registered pharmacist, a young scientist, an expert in the field of design and synthesis of bioactive hybrid molecules, computational drug discovery, with focus pioneering on hit to lead optimization associated with the development of novel bioactive ligands to fight against multifactorial diseases. Sir is a recipient of International Medical University Achievement Award in 2021 for teaching excellence and in 2019 for e-learning innovation. Sir is an innovator of technology enhanced learning strategies and a principal investigator of prestigious FRGS grant 2019 awarded by the Ministry of Education Malaysia. In International Medical University Learning Resource Festival Innovacy Competition 2020, Sir won a gold medal for his project E-Learning Innovation titled Flipped 180 Degrees, the partially flipped classroom model at IMU. In 2019, the Venus International Foundation recognized and conferred him as a young leader in health and medical sciences. Sir is currently a senior lecturer in Department of Pharmaceutical Chemistry, E-Learning Lead in School of Pharmacy, International Medical University, Malaysia. Sir has developed innovative technique, teaching and learning methodologies for the various course modules based on chemical education in undergraduate and postgraduate programs at the IMU, which helps students to enhance their learning experience. With this introduction, I request Sir to please address the gathering. Now the platform is open to you, Sir.
Thank you so much, Mrs. K. Vishalakshi Garu. It's a very warm welcome. I'm very glad to take opportunity to be participation, and also I have to congratulate Chaitanya University for organizing this educational webinar and sharing section. So without any further ado, I will be sharing my slides to discuss some concepts associated with drug discovery and development. May I have the confirmation? Can everyone can see the slides of mine?
Yeah, it's fine, Dr. Vasudev. You can proceed.
Okay, thank you so much, Dr. Koteshwara Garu. And so, dear participants, a very good afternoon to you all. I hope you all are very overwhelmed with the last two sections with good amount of information already grabbed and processing in your brains. So I just would like to make it this, keep it more concise and informative. Then, so let's start. Drug discovery and development, opportunities and challenges during COVID-19 pandemic. So just before we start discussing about the content of our presentation today, so just would like to remind you all, still we are in the pandemic situation. If you look at the number of deaths day by day still increasing. So please all of you take care of your safety and also practice all the SOP developed by the state and national health authorities. So what's the aim of this webinar? So in today's topic, I aimed at introducing participants with various opportunities and challenges related to drug discovery and development during COVID-19 pandemic. So intended learning outcome. So upon completion of this webinar, what's going to be the take home message for you all? So at the end of this webinar, you all will be able to acquire some knowledge on various adaptable strategies involved in the drug discovery and development pipeline. Moving forward, this is the content of my webinar. So I will be starting with the general introduction, followed by introduction to FDA approval process, types of simulation strategies, computer-aided drug design strategies, and COVID-19 drug discovery summary references, followed by acknowledgements. Moving on, let me introduce to you all the conventional drug discovery pipeline. So if you carefully observe the conventional drug discovery funnel, there are several stages associated with the drug, from discovery to development and development to the approval, and ultimately it reaches to the market at the stage of clinical use. So when you consider this whole process, the whole process is very typical process which involves different stages and multifactorial and multidisciplinary, which takes around 12 years of duration and $1 billion of R&D costs with no guarantee of success. So no guarantee of success is the key trigger where the R&D units are actually turning into a production units. That's the reason, because could not bear the loss. So the R&D cost is very high. Why? Because the development processes involves step 1 to step 2, step 3, step 4, and followed by until the FDA post-marketing approvals. So this is the total flow of the post-drug, the US FDA administration process. If you carefully observe the US FDA process, the preclinical trials itself takes 3 to 5 years, and followed by the clinical trials around 6 years, followed by NDA review, the new drug application review around 2.5 years. So in total, when you carefully observe, the estimated time is all around 12 years, and cost is also almost 100 million or 1 billion. Out of 5,000 drugs, only can 5 make it to the clinical trials, and then followed by the success of or failure, whatever it may be. So let us see some basic sources of drugs. The sources of drugs may be classified basically into natural, synthetic, and semi-synthetic. As you all know, the natural products are subclassified into plant, animal, mineral, microbial, and marine sources. So if you see some selective examples I provided for your recall and brainstorming. So this is not a lecture. This is mostly like interactive. So what I would like to discuss here is mostly to trigger the information associated with the new drug development processes. So the new drug development processes currently in COVID pandemic is not the conventional process, because if you see the conventional process, how long the time taking? It's around 13 years of time. And what's the expenditure? And what the manpower? Is it realistic to follow the conventional process at the moment? So it's very difficult because we are in emergency. So the need of drugs is day by day very high. So the survival rate of the patients who are affected with the or infected with the COVID, different variants are developing, and the resistance is developing for the existing compounds, which can boosting the immunity. So all these are major challenges associated with the COVID-19 pandemic drug discovery. So let us discuss about some strategies which can accelerate this process. So what are the strategies which can accelerate this process, and in what way they are actually accelerating? So some background also I will discuss, because you might be knowing the strategies very well, but what's the background? So background point of view, we need to understand some basic principles on what basis they have derived this particular technique as a complementary technique to support the drug discovery pipelines. So like that, just if you see a molecule from bench to the market, there are different strategies associated to bring the molecule from bench to market. So the basic steps involves the fragment based screening process. And so basically two things. One is high throughput virtual screening and high throughput screening. So what is high throughput virtual screening and high throughput screening? High throughput screening is the conventional process where a library of molecules will be subjected for a selected bioassay to identify a selective ligand that can produce a biological response. So that is high throughput screening. However, the high throughput virtual screening is the process of computational simulation where the outcome is just a hypothesis, but not a realistic ligand. So the hypothetical information what we are getting from the high throughput virtual screening must be subjected for the proof of concept study. So once you get the proof of concept study, then you could understand. So what is the realistic information you can carry forward? That is actually the transferable information. So this process, the high throughput virtual screening is a simulation strategy, but many of them having some misconception that simulation strategy can replace the conventional drug discovery process. Unfortunately, this is totally a misconception because none of the steps associated with the conventional drug discovery process cannot be replaced with a simulation strategy. Simulation can only complement the process by reducing the total number of experimental activities to be performed, but not the total replacement of the experimental activity. For example, let us take one small instance. You may need to screen 1 billion molecules in an R&D sector to identify one bioactive molecule. So instead of 1 billion, you can reduce down to the total number to few thousands or hundreds by applying some strategies. So what are these strategies? These are computer aided drug discovery strategies. So computer aided drug discovery strategies developed based on an information and data. So let us discuss more about it. So if you carefully observe this workflow, the first compound, which is a bioactive compound identified in the drug discovery pipeline, is a hit molecule. Once after you obtain a hit molecule, the hit molecule will be further processed to lead molecule. And this process is called hit optimization. And the lead molecule will be further processed to candidate molecule. And this transformation is called lead optimization. So when you carefully look into the hit optimization and lead optimization processes, there are several strategic filters associated. Filters. What are the filters? Filters are nothing but the standards, the quality parameters we are considering to enable the molecule attain desirable characteristics which a lead molecule consists. Similarly, the lead molecule will be further optimized to a clinical candidate with all the properties. For example, the best property you can consider. The toxicity alerts must be zero. No toxicity alerts must be there because preclinical studies is mainly to identify what is the toxicity profile with respect to the potency. So potency, nanomolar concentration, not even micromolar. 1 nanomolar, less than 1 nanomolar concentration could be identified as one best parameter to consider to optimize a lead molecule. And similarly, the permeation, absorption, distribution, metabolism, excretion, and toxicology. So every profile will be taken into consideration to optimize the molecule to the latter stage. So this whole process comes under lead optimization. So this is the total journey. It is really very iterative journey, dear participants. I'm talking about the various stages associated with the preclinical and clinical development. Okay. So what is the major challenge? If you carefully observe the growth of the expenditure, so total cost per approved new drug is drastically enhanced over the years since 1970s to late 2010. So if you see the total probability is only 11.8% as per the statistics. So such a very marginal probability, who can dare to put such expenditure in the R&D sector? It's rather very unusual to consider the R&D strategy as one of the strategic plan of the industry. That's the reason why the R&D strategies to adopt in different industries are day by day reducing, because the main problem is cost effectiveness. The total cost per approved drug is very high, and with no guarantee of success. And of course, successfully the drug is entered into the market. So in case if any major side effect noticed in the post-marketing surveillance, immediately the drug will be withdrawn from the market. So what about, just think about the company who put all the efforts and expenditure in the R&D sector. So this is the major challenge. So we must have some simulation strategies to integrate into the drug discovery pipeline to reduce the cost. The first thing is to reduce the cost. Okay. Moving forward, let us see what is a simulation. The basic meaning, as per the British English, a representation of a problem, situation in mathematical terms, especially using a computer. So especially using a computer. So simulation can be performed using a computer. In American English, use of a computer to calculate by means of extrapolation the effects of a given physical process. So based on these two definitions, what you could understand, you must compulsory, you must have a simulation data. So compulsory, you must have a realistic experimental data to perform simulation. So, dear participants, once again, the myth two, simulation can be performed without having any experimental evidence. So this is once again, we may need to relook into it. Simulation can only be performed if there is a realistic experimental method is existing. Why? Because simulation is the process of mimicking. Simulation means the process of mimicking a real scenario. So compulsory, you need to have a real scenario first so that you can mimic the process in the simulation. And what is the expected outcome? So the expected outcome is the simulation outcome and realistic methodological outcome both must tally. So this is what the validation. So if the results are comparable, the simulation strategy is valid. If the results are incomparable, the simulation strategy looks ambiguous. It's not valid at all. Because what is the purpose of doing it? You would like to reduce the total number of experiments which has to be performed using this particular equipment. So to replace that process, to complement that process, instead of performing thousands of activities, you would like to do hundreds of activities. So you are developing a simulation model. So simulation model must, the outcome of the simulation model must comparable with the realistic model. So that is the most important validation. So moving forward, let us see what is the landscape of simulation technologies. As you know that the ICT technologies are very well advanced. So if you carefully look into the domains of these simulation technologies, artificial intelligence, machine learning, and deep learning. So it all depends on how much intensity of parameters you are considering to develop your strategy. So based on that, the domains you can consider. It's a artificial intelligence is, for example, not only specific to the drug discovery, it is adaptable to across the industrial processes, the IR 4.0, Industrial Revolution 4.0, and cloud computing and Internet of Things. So these are all the digital disruptions, at the same time digital transformations taking place across the globe. So the artificial intelligence, machine learning, and deep learning algorithms are nowadays integrated into conceptualized chemistry and biology and macromoleculars and proteomics and genomics, so that the developed software-based simulation activities are complementing the pipeline-based activities of the real conventional drug discovery. So let us see what is the basic pillar of the simulation. So as I mentioned, simulation is the mimicking process. Simulation is the mimicking process. So what is the pillar? The simulation will be strong if you develop a simulation based on a real scenario, and the real scenario data will provide the information will provide the knowledge, and then to wisdom to take your own decisions, and the critical thinking and problem solving based activities can be adaptable at different levels. So data is the key information to be used to construct a simulation. So this data must be obtained from a realistic experimental data. So what is the real experiment you would like to consider to perform a simulation? Whether the real experimental model and the simulation model both are comparable or not. So I just would like to show you all a very nice flow diagram and how to validate the simulation model with the realistic model. So before moving forward to that, let us discuss about why do we need simulation. So the simulation, as we discussed, there are several stages. There are several stages in the preclinical as well, and from the clinical there are further several stages, and then proceed to the market to the drug will be approved to the clinical use. So in between the transition means moving from one stage to another stage. This is the transition. So in this transition, most of the experiments will fail. For example, I can give you one good example for the transition. Lead optimization to clinical candidate. So optimized lead molecule is ready at R&D level. But now this one must enter into the clinical trials. So the molecule may enter or may not enter. So what is this process? This process is called translational phase. This is the phase of translational where most of the experiments and ligands, compounds, new chemical entities will fail. So what is this process? The failure process is nothing but they technically termed as valley of death. Valley of death. So this is the valley of death. That is the meaning. So it is very difficult to transform from one phase to another phase in the whole drug discovery process. So this translational gap is the key gap associated. So how to address this gap? So how to close this gap? How to minimize the value of that possibilities and strategies? So this is what we need to address carefully. So generation of process of building a simulation model. So simulation model, as I mentioned, simulation is only possible if you would like to consider a real system. So the real system based simulation model will be more accurate and dependable. Why? Because what the real system is actually considered is actually taking lot of time to perform n number of experiments and lot of expenditure to procure the raw materials associated with the process and all. So what we can do is how to complement this real behavior with artificial intelligence behavior. So this is the whole process of designing and developing the algorithms. So real model, develop a model, make a model. From the real model, starting point is the real model. Make a model and perform experiments from the real model, and experimental results will obtain. Okay, then followed by, if you see the model system, perform simulations as well, and simulation results will be obtained, and construct approximate theories and theoretical predictions, simulation results, and experimental results. When both all the three different types of outcomes are in the same alignment or in the same platform, so where they are comparable and consistent to each other, at that point of time, you could say that the simulation model is developed and also validated. So in case if any discrepancy observed in between the values obtained from the experimental as well as simulation and theoretical predictions, so meaning to say that the developed simulation model is not validated. So this is the place where most of them, they generally don't consider this particular process when they are performing simulations. So that is the major problem. Otherwise, if you carefully look into the Google search, just press COVID-19 drug discovery, thousands of mushroom growth of articles publishing with the simulation models. And why so far we are struggling to secure a one FDA approved selective drug to fight against COVID-19? Because no specificity. Domain of applicability is poor. Validation statistics are not applied. So all those things. So when you are talking about a simulation model, compulsory the validation criteria, the cross validation or normal validation criteria must match. After, if you consider this is a robust model, it is a reliable and realistic to adapt in the industrial environment, then that is the real achievement. Okay, this is how the process, general process of building a simulation model. But before performing a simulation, I am sure majority of the researchers, the budding researchers, mostly who are actually emerging in the field of drug discovery, they are very enthusiastic. They would like to do something new. However, some gap in their particular understanding or competency, this is generally called as competency gap. If you ask me some details much about the pharmaceutics, I may not able to express properly because there is a competency gap. My specialization is different. So like that, due to that competency gap, they may ignore few steps associated with the simulation, which are very important. However, so let us move forward to discuss about what are the various technologies can be integrated into the drug discovery and development. So if you carefully observe, these are the standard pipeline is the standard process, meaning the conventional process. None of the stage in the conventional process can be replaced. So if you see the point arrow, the arrow point indicating different stages, they are all complementing the processes. These are the strategies associated with computer-aided drug discovery. So various tools are available. So I'm going to introduce few tools to you all. So just see and enjoy. So moving forward, these are the application point of view. So this analogy everyone knows, lock and key. At this point of time, I would like to recall one of the meritorious scientists who contributed to the field of chemotherapy and also the pharmacology, the term receptor coined by Paul Ehrlich. At the same time, he's the father of chemotherapy as well. So this great scientist, he was the one who achieved the Nobel Prize in the year 1908, Paul Ehrlich. Clearly, hypothesis is that drug will only produce its response unless it binds. So without binding to a specific receptor, it could not able to produce any kind of response. That's what the hypothesis generated and well validated by support of various receptor theories. We can see the Mayers-Overton theory, so many theories actually later they developed. So once just we directly move on to the accelerating strategies related to the drug discovery, the simulation integrated scaling processes in drug design. Why I put scaling process? Because based on the simulation, we consider the outcome to do the real experiment. So for example, you have billion molecules. You are not interested to screen in the normal conventional way. So you just applied a ligand-based strategy to screen the database and obtain few molecules, few hundreds of molecules, and then proceed to the experimental process. And then you may identify a few hit molecules. These molecules may be further optimized to lead and further optimized to drug candidate and so on. Okay, so this is a general workflow. So now let me start with basically, so in this general workflow, if you look at the strategy, basically two things. One is structure based, the other one is ligand based. As I mentioned to you all, in the beginning, simulation starts from data. So data and followed by information and knowledge and so on. So in case the information of the protein drug target is known, okay, then we can consider structure based drug design. In case the ligand based drug design, if you would like to opt, in generally we will consider only the ligand with known chemical formula, but the mechanism, the exact mechanism of action unknown, the selective target binding site unknown. So in such case, we just follow the ligand based drug design strategy. So if you start with the structure based drug design, mostly protein-protein interactions are possible, protein-DNA interactions as well possible. So if you consider these protein-protein interactions, we can consider the COVID-19 already, because COVID-19, the virus is directly attacking to angiotensin converting enzyme 2. So this is where the protein-protein interactions also taking place. Which protein of the COVID-19 is actually interacting with the angiotensin converting enzyme, which is the spike protein. So that's what we are going to discuss in the next few slides. So DNA ligand interaction. Hope you all are from the pharmacy background. You know definitely what are alkylating agents. Alkylating agents, potential anti-cancer agents, first synthetic chemotherapeutic agents identified in that field. So this is how the DNA alkylation of the DNA generally takes place. and the conformational isomerism and the receptor flexibility, induced fit mechanism. These are all the protein ligand interactions will generally takes place as well. So structure based drug design simulations are basically consists of different strategies. This is one such strategy as well, molecular docking simulation. And moving forward to the homology modeling. If you look at the homology modeling, what is the basic fundamental principle of doing homology modeling? So homology modeling actually refers to construction of atomic resolution model of the target. So for example, not every isolated sequence of the target protein can be constructed to a three-dimensional X-ray diffraction crystallographic structure, because the X-ray crystallographic structure needs high stability and also withheld of the whole of lot of pressure in the cabin. So X-ray diffraction is not so easy to perform to every single isolated target protein. So sometimes we may not able to get the crystallographic protein. In such case, homology modeling is the best strategy to adopt. So what is homology modeling here? For example, if you look at the primary structure of the protein, what is the primary structure of the protein? An amino acid sequence. So this amino acid sequence may have some close structural similarity with the sequence similarity with any other amino acid sequence. So the second one. So this is the template sequence and that one is the query sequence. So in that point of view, you may be having some sequence similarity between the two amino acid sequences. Based on the sequence similarity, if in case the query template, the query sequence consisting of already three-dimensional structure, because of the sequence similarity, you can adopt this sequence into alignment of the template modeling, so that a three-dimensional model will be developed. So this is artificial model, but not X-ray diffraction realistic model. But still, since the similarity in the sequence is close to each other, so there will be a possibility to exhibit the same biological functions as well. So that's the major concept integrated into this technique. However, there are several problems associated with homology modeling. Why? Because the Ramachandran statistics, the quality of the protein will only be confirmed based on the Ramachandran statistics. So in the Ramachandran statistics, if any amino acid deviates from its dihedral angles, so that one will be considered as a defective protein. So in such case, we may not able to consider that protein for doing the fragment based drug design or structure based drug design. So moving forward, the next one is fragment based. So this is what the current strategical approach we adopted to the COVID drug discovery. Why? Because the flexibility within the binding site of coronavirus targets not enabling any one of the researcher to identify the specific ligand. So the specificity of the ligand is varies from strain to strain, varies from the binding site to binding site. At the same time, allosteric binding sites are responsible for this process as well. So the selectivity and specificity becoming very, very challenging to handle at the R&D level to find the new drugs to fight against COVID. Okay, so if you see the small fragments will produce small biological activity. So by incremental construction, by selecting a small compounds together, you can, this is actually considered as building blocks. By building blocks, it's just like you can consider one block and connect it to the next one and connect to the, so that it will improves the selectivity within the target site. So this is how the target site information can be further modified. So the selectivity, if you carefully observe in this case, 1 micromolar is the initial the fragment. Upon successful incremental construction, you can see 10 nanomolar. So that is how the potency will be also directly correlated with the lead end of the compound as well. Okay. So molecular docking simulation, this is well established strategy to identify a ligand. So why? Because molecular docking strategy only be performed if you have the complete information about the receptor target, the target receptor, and also the co-crystallized ligand. So if you do not know the co-crystallized ligand, you may not able to perform the molecular docking. Why? Because the co-crystallized ligand will produce the clear information to you to extract the binding site coordinates. So the binding site coordinates, if you see in the simulation, the molecule is entering into the specific pocket of the protein target. So what is that specific pocket? The specific pocket is nothing but binding site region. So the binding site region where the molecule is going and binding, that's very important to understand the nature of the binding site and to understand the orientation of the ligand and to understand the types of binding interactions generally taking place within the binding site. So what are the different types of binding interactions? You all know very well. Since beginning of the pharmacy course, we come across with various chemical interactions like induced dipole interaction, hydrogen bonding interaction, and van der Waals interaction, hydrophobic interaction. These are all various types of interactions. So drug target binding forces are key forces actually. So in order to take those forces into account, you need to have a specific ligand. And if you ask me, how can we say these? Are there any successful stories? All the HIV protease inhibitors, all the reverse transcriptase inhibitors, swine flu virus, and neuraminidase, zanamivir, all these potential drugs entered into the market only based on the structure-based drug design through molecular docking strategies. That is the acceleration made by Human Genome Project. Because of Human Genome Project, a lot of advancements, especially in the genomics and the proteomics, actually accelerated the whole process of drug discovery. So this is how about the target plus ligand produces the molecular docking, and the binding energy calculation is the key component here. Binding energy means before compound binding, what is the thermodynamic energy of this particular target protein, and after binding, what is the total energy of the protein ligand complex. So what is the difference between the before and after binding? So this will produces the total enthalpy. So this is the thermodynamic system where it can calculate by the designed algorithms. So you may be surprising to see that how these kind of softwares are basically generated. So the software is basically developed with two major fields. One is molecular mechanics and the other one is quantum mechanics. So these are the integrated fields, deals with the atomic physics and empirical methods associated with the development of algorithms. These empirical methods are mathematical formulas. So the mathematical formulas easily can be adopted into algorithmic language. There are several computational languages are there. So you can see Python and BioPython, different SQL, different types of languages are there which are embedded neuronal network based frameworks and so on. So these are some of the strategies as well for the structure based drug design. When it comes to ligand based drug design simulations, so ligand based means what is the information we have? We have only the ligand information. So what is the exact mechanism of paracetamol? So the exact mechanism of paracetamol not at well established. Do you know, students? Dear participants? No. If you just press drugs with unknown mechanism of action and open the Wikipedia page, you can see n number of compounds. So far the well established mechanism is not elucidated, but still we are using in our day-to-day life. Why? Because they are safe and efficacious. That's all. And there is a therapeutic benefit. So just because of that we are using, because whatever you are administrating and how much the percentage you administered and how much is excreted and eliminated from the body. So if both are tallies, okay, there is no harm. As long as the compound is safe and efficacious, exhibiting some therapeutic response, you are good to go. So that's what the strategy adopted. But well-established specific mechanism on by which the particular compound is exhibiting its response. So not well-established. Many drugs, even many drugs. So the basic ligand-based drug design concepts are categorized into three. One is ligand-based pharmacophore modeling, ligand and similarity, and 3D QSR modeling. So coming to similarity search at the beginning. Similarity search is at the atom level. So this is the initial starting stage of identifying atomic level. So all the atoms will be taken into consideration to calculate the similarity. So when it comes to the atomic level, if all the atoms are taken into consideration, what is the main advantage and what is the main disadvantage? So advantage is closely structurally similar molecules will come as the outcome. Okay. So what is the disadvantage? It will be only limited to the chemical diversity of that particular compound. It cannot expose the different diversified molecules which are also present with similar type of functionality. So later on, this similarity set has been modified to pharmacophore modeling. Pharmacophore is also considered similar to the similarity, but the information what they generally consider is the steric and electrostatic features. The basically all chemical classes of compounds categorized under 6 different classes. One is hydrogen bond acceptor, hydrogen bond donor, positive ionization, negative ionization, aromatic ring system, and hydrophobic system. So these are the various types of functional processes we generally come across. Okay, so pharmacophore modeling is the process of selecting a suitable functional moiety. That functional moiety may be a hydrogen bond acceptor, donor, or any other source. So the mapping of these functional groups, these functional features, chemical features in a molecule will produce as 2 different parameters. So if you connect more than 2 points in a molecule, the 3-point model will produce as 2 different parameters. What are those 3 different parameters? 2 different parameters. One is interatomic bond distance. The other one is interatomic bond angle. So if you connect a 3-point model with suitable pharmacophores, first of all, what is a pharmacophore? Pharmacophore is the chemical functional moiety which is responsible for its biological activity. So how do you know it is a pharmacophore? For example, based on the structure activity relationship of the lead optimization, you could understand. So if you remove this ring system, the molecule suddenly devoid of activity, drastic downfall of the activity. So meaning that this is a pharmacophore. You should not remove this compound. This particular fragment is very much essential in this compound. Why? Because this is a pharmacophore. Why? If you remove this particular one, the whole activity will be affected. Okay, so in that aspect, you will identify few pharmacophores and develop a pharmacophore model. And this model can be subjected for the screening based on the 2 parameters. One is interatomic distance and interatomic bond angle. However, the parameters can be further advanced to inclusion of both the distance parameters as well as the functional moieties, like for example, the first one is hydrogen bond acceptor, second one is donor, and this is the ring system. So a 3-point model, acceptor, donor, and ring system. So this will produce a number of hit molecules. So these hit molecules are ready to proceed further to the laboratory experiments. In the laboratory experiments, if you see any good hit molecules with desirable properties, so the pharmacophore model has been successfully validated. So this model is specific. This model is very selective to identify what set of molecules. That is what domain of applicability. So this model can able to produce what? Yes, I can say like this. This model can produce a potential anti-cancer compounds. This model can produce a selective alpha glucosidase inhibitors. This model can produce a selective H proton pump inhibitors, like that. So what selectivity you have based on the selectivity of the pharmacophores and from which the bioassay you derived this compound, and say all those factors are influencing this particular selectivity. Okay, this is pharmacophore modeling. Moving on, QSAR modeling. When it comes to QSAR, this is one of the well validated and industrially adopted technique. So dear participants, if you ask whatever the strategies we discussed, or we have any specific regulatory requirements for those? No, there are no specific regulatory requirements at all, because all are imaginary, all are hypothetical. Most of them are not reliable, but QSR is not like that. QSR is a reliable model. However, QSA are also having a specific OECD guidelines, Organization for Economic Cooperation and Development, OECD guidelines, same like our pharmaceutical method development and validation. QSAR also has its own method development and validation guidelines. If your developed model successfully passed all the guidelines as per OECD, then this model will be adopted to the industrial scale as a primary filter to screen the database of compounds, to identify the hits to carry forward to the experimental studies. So this is how they can generally proceed. So QSR is very well-known model since beginning. We also know the log p value, the initial analysis of the Hans analysis, where the log p partition coefficient directly correlated with the observed biological activity, and Mayer's-Waverton coefficient, Waverton theory also directly correlated with the observed general anesthesia with the lipophilicity and so on. So there is a long history starting with Corwin, Hans and so on. So it is a mathematical model. So this mathematical relationship, since the mathematical theories are proven, so this is a adaptable model. So more strategical. So now moving forward, dear participants, so after discussing all these strategies, what is your opinion? Do you think are they have any specific limitations for the computer-aided drug design, or everything is very perfect and we can readily adopt all these strategies to perform new drug discovery? There are few limitations. So why? Because if you critically appraise the limitation, what you are performing, what you are doing, you will have clear idea. If you overcome this limitation with closing one eye, so automatically whatever you are doing is a very hyphenated research, you feel like that, and you just feel yourself as a very high-end research is going on because you closed one eye already. You are not looking at the other side of the perspective, which are limitations of the computer-aided drug design. So limitations are a realistic endpoint. So what is the endpoint after performing this particular simulation? Whether the endpoint is matchable or not. And simulation, and the next one, an ambiguous algorithm. So ambiguous algorithm, for example, you are giving input and you are getting the output. Okay, you are just putting your entering some details and giving the input and outcome. What is the exact process of doing this process? How come I getting the outcome? Of course, it is an intellectual property. At least to the authorities, you must clearly mention these are the logical steps involved, and the logical construction of the algorithm is based on. An ambiguous algorithm is always a troublesome to a researcher, because researcher may not able to rely on that algorithm because the complete details are never established or disclosed to the authorities. For example, if you consider selecting a topmost drug discovery company who actually providing services, and unfortunately you used all the tools and you managed to identify few hit molecules, but none of the experiments producing any success, not even a single experiment is successful. So all your proof of concept activities are failed, maybe because of this ambiguous algorithm. So this is very important. And defined domain specificity and applicability. So this is the most important we need to discuss about. Domain specificity and applicability, meaning to say that which biological activity you are targeting this particular simulation process, and what is the outcome of this simulation process, in what way it is useful to transfer to the next level, and what is the diversity, meaning that it is applicable to adapt to how many types of strategies. So the applicability and specificity must be very clear, so that the strategic flow will be successful at one or the other time. So next, appropriate measures of predictive power. Appropriate. So whether it is a measurable or not measurable. For example, I show a picture with a very good orientation. So how can you measure that? So only if I take the photograph and send to you, could you able to detect what is the exact orientation, what is the root mean square deviation within the binding site region, And what is the exact conformational energy of that compound? No, not possible. So we may need to have some outcome measurements. Outcome measurements is one of the most important tool to identify and compare. So if you don't have the outcome measurement, how do you compare? How can you say that this is prioritized, this is non-prioritized? How can you say that this is ranked 1 and this is ranked 2? Not possible. So compulsory, the appropriate measurement should be there. So the measurement may be the binding energy, the measurement may be the total number of hydrogen bond interactions, or the measurement may be the solvation property within the binding site region, and so on. And then finally, a mechanistic validation. This is the major problem. Mechanistic validation means what is the real experiment? This is what we already discussed. I'm not going to go into much details. So a mechanistic validation means what is the experimental activity with is that comparable with the virtual simulation activity or not? That's all. That's what the mechanistic validation. So these are all the limitations associated. So with these limitations, do you think the drug discovery program is really accelerating anti-COVID drug discovery in the current scenario? Are we able to get the drugs on time? So people are dying. So day by day, the number of deaths reporting to the WHO are almost few millions. So it's 0.4 million already. So if you consider which strategy is the most suitable strategy, you think, dear participants? So we may need to embark on a strategy which is so realistic, is really strategic, and also can bring the product to the market at the earliest. So that's what the real strategic plan. This is not a real strategic plan. This is the conventional integrated with the accelerated drug discovery program. But just imagine whether you could manage to get the compounds on time or not. So let us move forward. So the strategy what we are talking about must bring the compound directly into the market, and it must take very less time when compared to conventional process, and so on. Drug repurposing is the best strategy to adopt at this pandemic situation. Drug repurposing. What is drug repurposing? It's well-known strategy, identifying new uses for the old drugs. Identifying new uses for the old drugs. Drug repurposing. So, for example, a drug molecule which is already existing in the clinical use, why don't you take that same molecule and do the COVID-19 screening in the biological laboratory? That's all. In case if the compound is active, proceed further to go for the clinical trials and so on. Why? Because the drug has already received safety clearance, efficacious clearance, acute toxicity, and all preclinical filters actually crossed. Drug-like properties are there, lead-like properties are there. So compound is complete already. Only thing is we may need to see what is the specificity towards the COVID virus, and what are the various target selective activity observed in the COVID activity, so on. That's all. So this is what they started with in the mid of COVID pandemic last year, when COVID-19 started immediately. So there was a huge acceleration when it comes to the marketing strategies that hydroxychloroquine has been identified and repurposed for the COVID-19, and later on it has been withdrawn because of some side effects and so on, not very less or not very specific towards the COVID virus. And then finally they managed to identify a very potential drug. Certainly that's the only compound we managed to get till date. The only compound is remdesivir. So remdesivir is also one of the best fruitful outcome of drug repurposing. So do you think drug repurposing strategy is suitable for our current pandemic situation or not? So just imagine. So drug repurposing, why it is so useful? Because the only thing is the development time is very less. At the same time, the chances of failure is also very less, because almost the safety profile, everything already, plasma absorption, distribution, metabolism, excretion, toxicology, well known. So Of course, I can give best example to you all. Initially, the aspirin used as a non-steroidal anti-inflammatory drug. However, it is one of the emergency indexed medicine in all the cardiovascular diseases as a potential anticoagulant, antithrombotic. So that's what the drug repurposing comes from. So drug repurposing is the current strategy at the industrial level. Mostly they are focusing on to identify new drugs to fight against, especially in this kind of pandemic situation, because we don't have any other alternative to start the drug discovery process and development at this point. Maybe in 2035 we maybe end up with a drug molecule. By that time, what is the, we could not anticipate what could be the situation of the global population. Maybe by 100th population also, maybe, yeah. So we could not anticipate what could be the circumstance at that point. So let us see the advantages. You can see the timeline drastically reduced, almost come down to 1 to 2 years. But still, so if you ask me, so it's all around. It, although it's identification, everything is processing, accelerating, but still taking its own time, around 6 to 8 years. Then how come already the compound is entered into the market and using for the clinical purpose? So this is one of the FDA initiative. So what is this kind of initiative? So many of the think, okay, just question to yourself. So we may need to go through all these steps in order to have the molecule to enter into the clinical use. But why the molecule is already available in the clinical use and currently we are using? Did the really the sufficient time has been studied? No. Sufficient time not studied. Why? Because of emergency. So what is this initiative? So let us discuss about the coronavirus, some structural activity point of view. I'm not going to details about molecular biology and so on. Just would like to introduce to you all the possibility of targets from the coronavirus. So this is actually coronaviruses is ranging from non-pathogenic to severe pathogenic infections. So with like basic common cold and to the SARS coronavirus. The major drug targets are main protease, spike protein, and RNA-dependent RNA polymerase. These are the three major targets. Same like if you refer to the HIV infection and Ebola infection, and almost a similar type of family. So reverse transcriptase and HIV protease, integrase, similar type of same type of protein drug targets also present in this enveloped uh, virus. So this one, the main protease, if you see, this is the main protease is actually very, very important. Why? Because the functional proteins of the coronavirus will be synthesized by the main protease, because the main protease is the one that at various amino acid connections, it will actually cleave the amino acid chain to produce a functionally, structurally stable virion particles. So these particles are form part of the internal integrated components of a coronavirus and synthesize a matured virion. So main protease is the one could be a special target to design a novel molecule. Okay, so attractive target for design a novel molecule in what basis? Because we already able to manage to identify the structure and the therapeutic, the three-dimensional target and what is the selective binding, but still could not able to identify a selective ligand with more potency at nanomolar concentration. Why? Because due to the conformational isomerism of the compound binding, the target binding site. The target binding site is very flexible, and due to this flexibility, it attains different conformations, and not only conformation, this due to the mutations in the binding site amino acids, different types of variants are generating. So these different types of variants are not specific to the ligand what we design. So these variants are basically developing a lot of resistance when compared to one to the other. Okay, so moving forward, the challenge to design a specific ligand, you can see here. So shown the protease of viruses can be very different. So the drugs designed to fight one may not be effective to others. So whatever the drug we design to specific variant, so that particular variant only be possible to inhibit that particular catalysis. But in case if the variant managed to change its form, So certainly this molecule may not be show any kind of activity because that process of resistance has been developed. So the spike protein is the one which is the main protein responsible for initial interaction and the inducing the virion into our human host. This is the main one because the primary target of antibodies as well. And the crown-like appearance is the one. This crown-like appearance is the key responsible structural feature that is very flexible, and the domains consisting on the spike are very conformationally flexible, and the domain is actually consists of the sugar backbone, and this sugar backbone, all the bonds are very flexible. So it will not attain a specific stable conformation at one point of time. So it may have once again different flexibilities. So once again, this is the major cause for that could not able to identify a specific inhibitor for the spike protein as well. So you can see here the flexible features. Flexibility is one of the greatest advantage to the virus, not to the human host, not to a researcher. So because of this flexibility, recent variants are virulent because their receptor binding domains are very flexible. Flexible means the flexibility is because of the amino acids and the rotation of the bonds. Okay, where the more widespread coronaviruses cause common cold and less dangerous because of they are less flexible. So common cold also causing due to the coronavirus, but coronavirus is when compared to the SARS coronavirus 2, why the common cold causing coronavirus is very less infective and less pathogenic when compared to. Why? Because these spike proteins are less flexible. So because of that, our antibodies will immediately can grab that particular target protein and fight against coronavirus and so on. So moving forward to the next one, the receptor binding is the phenomenon how it binds to the angiotensin converting enzyme 2. So is the one responsible, and the target binding site is angiotensin converting enzyme 2. And this angiotensin converting enzyme 2, based on our background, what is the most important target is this one. This is actually an antihypertensive drugs because ramipril, eosinopril, all those compounds. So this angiotensin converting enzyme is the key target site which is responsible for the receptor binding of the spike protein. Okay, so this is what making these cells target for the virus. So next one is RNA dependent RNA polymerase. This is going to be very special target. Why? Because the process of human genetic system is totally different from the RNA dependent RNA polymerase. So because this process is not existing in the human, the single strand RNA and development of DNA to RNA and so on, this process is totally different. So comparable to the RNA, the comparable to human host, this RNA dependent RNA polymerase based activity is more specific to the ligand. So that is how we actually manage to focus on drug discovery to a specific ligand binding domain in the RNA directed RNA polymerase. However, but still this compound is also having some limitations, but the compound identified was remdesivir. And remdesivir is not a selective compound specifically identified for the coronavirus. Remdesivir initially identified as a potential Ebola, anti-Ebola compound, but still the anti-Ebola activity is not so promising. So for that reason, they actually repurposed this compound again towards the coronavirus because both are dealing with the viral particles and the specificity probably the same. And once again, the remdesivir is not a drug, it is a prodrug because the active forms of the drugs is almost follows the similar steps associated with acyclovir. So monophosphate, diphosphate followed by triphosphate, and then complexes, it is a competitive inhibitor as well. Because why it is a competitive inhibitor? It is basically a adenine based compound. So you can see here. So next one. So these many things are going on across the globe. So what are the FDA initiatives? Because Food and Drug Administration US is the key operator across the globe, the medicines and approvals to all those things. So FDA initiatives also accelerated, and this is the current statistics as of today. So 630 plus drug development programs in planning stages, and trials reviewed so far are around 460, and 11 COVID treatments currently authorized for emergency use, treatments currently approved by FDA for use in COVID. So you may be surprising between these two, emergency use and approved use. So Remdesivir is an approved. So what is the emergency use then? So you might be confused between these two. So I just would like to show to you, emergency use is nothing but also studied partially, but due to the pandemic. So according to 564 of Federal Food Drug and Cosmetic Act, some compounds will be authorized to use in specific conditions like pandemic situations or something like taking decisions. I don't want to review the compounds here because there may be some discrepancies or regulatory affairs may be involved. That's why I just would like to bring to your notice that EUA drugs means emergency use authorization drugs. They are not completely studied and passed through all the phases of compounds, but they are actually approved for some other use. But to produce some symptomatic relief in COVID-19 patients, especially for inflammation or immunity boosters or something like that. So in such case, emergency use authorization is the one responsible to adopt and then proceed further. So this is one difference, the approved thing and also emergency use. So what's the best way to fight against COVID-19? How to avoid COVID-19 spread? So after reviewing all these strategies, and we discussed this one as part of the knowledge sharing on what are the current practices of drug discovery and development and what best strategies to be adopted at this point of time during the COVID-19. We already come to a conclusion that drug repurposing could be more suitable at this point of time. Why? Because we are rushing to identify the new molecules as well. So what is the best way to fight against COVID at individual perspective? Just act. What's in your control? What's in your control? Just do cleaning, sanitation, and when you do the sneezing or cover this with sufficient equipment and healthy hands, face mask, physical distancing. Feeling sick, just stay home, don't go to office. So it is totally our responsibility. So the social responsibility is equally important than the identifying a new drug molecule. So identifying a new drug molecule is totally hypothetical. It may be realistic at one point of time, it may not be realistic. So it is totally hypothetical. But practicing the good protocols, the SOPs and safety measures and maintaining the social, this all in under your own control. So please take care of yourself. Don't depend on any other person. Take care of your individual responsibility. Start at your own and adopt at your home. Automatically, it will be spread across the society, and the national health policy, national health status will be upgraded. So please, all of you, stay safe and take care of you and your family members and the nation. So in summary, computational simulations have played major role. Good number of successful stories are there. Several FDA approved, also designed and intensively used strategies to identify the lead compounds. But what is the real breakthrough in simulation is that in our discussion point of view, when you perform a simulation, the moment the outcome is obtained from the simulation, that's it. So no more further cross checkings or validations required. So this one directly carried forward to the next experimental study. So that is the stage where we can say that we are very much advanced and integrated technology more effectively, overcoming all the barriers associated with the conventional and traditional processes of the drug discovery. So this is what I would like to end my presentation. And our take home message is keep calm and stay safe. These are the references I used to construct the slides. So My acknowledgements is to Associate Professor Dr. Muhammad Julkifli, Dean School of Pharmacy, IMU Malaysia, for providing all the approvals to participate in this sharing section, and equally Associate Professor Dr. Kamsuki, Associate Dean Academic, School of Pharmacy, IMU Malaysia, Professor Mallikarjuna Rao, Associate Dean Research, School of Pharmacy, IMU Malaysia, Dr. Lee Choi Sin, Head of the Department, Pharmaceutical Chemistry, School of Pharmacy, IMU Malaysia, and Professor Dr. Y. Rajendra Prasad, Principal and Professor, AU College of Pharmaceutical Sciences, India, my supervisor and mentor. Dr. CHV Purushottam Reddy Garu, Founder and President and Chancellor of Chaitanya University. Professor Damodar Gurappu, Vice Chancellor, Chaitanya University. Dr. K. Veera Venkataya, Officer on Special Duty, Chaitanya University. Professor M. Ravindra, Registrar and Chaitanya University. Dean and Associate Deans, Head of the Department, School of Pharmacy, Chaitanya University. Associate Professor Kumaraswamy, Organizing Chair, and beloved participants. Dr. G.S.N. Koteshwar Rao, Director, Maibo Group, Press and Media. Thank you all so much. So, any questions?
Okay, thank you very much, sir, for your wonderful talk on drug discovery and development, current opportunities and challenges during COVID-19 pandemic. Thank you for enlightening us with the insights of this process of drug discovery and development. Thank you very much, sir, for discussing these strategies involved in the journey of a molecule from bench to market, which will help the young researchers who are working on this, bringing a newly synthesized molecules to the market. Thank you very much,
Thank you
sir,
so
for
much,
sharing
ma'am,
this
for facilitating
information
this
on
section.
this drug repurposing, particularly its examples, HCQ, and its applications, all this, sir. Thank you very much, sir. Over to Divya, ma'am.
Thank you, Mrs. Vishalakshi Garu. Now, on behalf of Chaitanya deemed to be University, I would like to request the invited speaker, Dr. Vasudeva Rao Avpati Garu, to receive a memento as a token of honor.
Thank you. Do I need to
Thank
take
you.
any
Do I need
action
to
now?
take any
I
action
need
now?
to stop
I
sharing.
need to stop sharing. No, sir. One second. Okay. Thank you, sir, for sharing your immense knowledge to this scientific platform. Now you can left the session, sir. Thank you so much once again.
Okay, thank you so much.
Okay, thank you so much. We have now come to the closing session of today's webinar. I strongly believe that the participants got enriched their knowledge in the area of pharmaceutical sciences with such a wonderful and valuable informative sessions by the eminent speakers of excellent expertise. And now I would like to preside the event to the organizing secretary of today's international webinar, Dr. G. Kumaraswamy Garu, to speak on this occasion. Over to you, sir.
Thank you, Divya madam.
Thank you, Divya madam.
Good afternoon, one and all.
Good afternoon, one and all.
Myself, Dr. G. Kumaraswamy,
Dr. G. Kumaraswamy,
Faculty of Pharmacy,
Faculty of Pharmacy,
Chaitanya deemed to be
Chaitanya
University,
deemed to be
Warangal,
University, Warangal,
and organizing
and
secretary for
organizing
the
secretary for
present
the
international
present
webinar
international webinar
on emerging trends in pharmaceutical sciences. I am very much thankful to all the registered participants for showing interest in knowing the current trends in our field, and happy for the overwhelming responses across the globe. And the intention of our webinar is to let the participants gain some additional knowledge in some premier sector like a pharmacovigilance, entrepreneurship, and drug discovery and development. And the speakers have really given wonderful information with their expertise in the respective field. I thank to all the speakers for their valuable time and commendable talks. I hope the participants gain sufficient information and the wish of Chaitanya deemed to be university in organizing this webinar has been accomplished thanks to our university authorities and Department of Pharmacy and permitting us to plan and execute this event. I'm also happy to organize this association with MIBO Group and IPA, Indian Pharmaceutical Association, Warangal branch. Okay, next, I requested to Dr. Chandana Madam, Faculty of Pharmacy, give a vote of thanks. Over to Chandana Madam.
Live participation of this international webinar on emerging trends in pharmaceutical sciences organized by the Department of Pharmacy, Chaitanya deemed to be University, Warangal. It's a great privilege to render vote of thanks on this occasion. On behalf of my institution, I would like to thank each and everyone who have directly and indirectly involved in making this international webinar to be organized. Firstly, I express my sincere gratitude to the chief patrons of the event, Dr. CHV Purushottam Reddy Garu, Chancellor, Chaitanya Deemed University, and CH Suvarna Devi Garu, Secretary and Correspondent, Vishwa Bharati Education Society, for giving us an opportunity and immense support to organize this one-day international webinar. Thank you, sir. Thank you, ma'am. I thank the patrons, Shri E. Vikram Reddy Garu and Dr. C.H. Satvika Reddy, Vice Presidents of Vishwa Bharati Education Society, for being a part and gracing this webinar with their presence. My sincere thanks to our chief guest, Professor G. Damodar Sir, Vice Chancellor, Chaitanya Deemed University, for his presence and valuable suggestion in conducting this international webinar. A special thanks to our guest of honor, Dr. K. Veera Venkataya, Officer on Special Duty, Chaitanya Deemed University, and also Professor M. Ravinder Sir, Registrar, Chaitanya Deemed University, for providing their support and guidance to make this webinar a grand success. Thank you, sirs. I would like to thank Dr. A. Pramod Kumar, Pharmacovigilance Expert and Drug Safety Officer, for enlightening us with his talk on overview of pharmacovigilance in India. Thank you very much, sir. I must mention my deep sense of appreciation for Professor Dr. Satish Babu Natarajan for his explanation on booming entrepreneurial opportunities in herbal industry. Thank you very much, sir. Further, we are grateful to Dr. Vasudev Rao sir for demonstrating his talk on COVID-19 drug discovery and development, current opportunities and challenges. Thank you, sir, for your wonderful lecture. I must thank Dr. V. Mallikarjun, Dean, Pharmacy Department of Chaitanya Deemed University, for his enormous cooperation in organizing of this event. Thank you, sir. I would like to take this opportunity to place on record our hearty thanks to Dr. Kumaraswamy Gandla, Organizing Secretary, and Dr. Divya Ajmera, Coordinator of the event, for their planning and effort in bringing all the esteemed delegates on a single platform through this international webinar on emerging trends in pharmaceutical sciences. I extend my thanks to Indian Pharmaceutical Association, Warangal branch, for their association, as well as Dr. Koteshwar Rao Garu, my Director of MIBO Group, for their technical support in making this virtual meet a grand success. Finally, I congratulate and thank all the students for your active participation in this international webinar, and all my dear staff members of Chaitanya Deemed University Pharmacy for your cooperation and support and bringing this event a grand success. Thank you one and all. Have a good day, and over to Dr. Divya.
Dr. Chandana. Thank you, Dr. Chandana. I, Dr. Divya Ajmera, on behalf of Chaitanya DMTB University, would like to thank respected distinguished dignitaries, guests of honor, eminent invited speakers, and all the participants from distinguished disciplines for your participation and cooperation in making this virtual event a grand success. I further take this opportunity to appreciate all the participants for your valuable positive feedback, which will boost our enthusiasm in conducting more scientific events further. Thank you, one and all.